Do you see the entire screen or just the presentation?
We see the slides across the bottom as well. We see the presenter mode.
Okay,
There you go.
there we go. All right, well, welcome everyone. This is a great moment each year, and although we're doing it a little differently, it's no less of a momentous day to be at our 24th annual research day for neurosurgery and the 18th annual Ved P. Sachdev Memorial Lecture. That's a lot of time and a lot of years. The agenda today will include my introduction, and I'll introduce both the Sachdev Lecture and the Research Day and the Research Award concept. Then we'll have the Sachdev Lecture, the research presentations, and then a new Research Day presentation winner. So I want to start with how this all came about. In 1996, a few years after Cal formed the new department and after I joined and Isabel and Chandra Sen joined, with Wade Sachdev here and Lenny Malice here at the time, Isabel came up with the idea that we would have a basic science day, since we were focused so much on clinical medicine and growing the program in those early days. But our emphasis also was on science and research. And so she organized a Grand Rounds day. It was a half a day, as I recall. It was called Basic Science Research Day, and that grew over time. At one point, it got so big that we had parallel sessions. We had a full day. It became a really big, long, and very deep day, underscoring the research that was being done in and by department members. The pendulum has swung to and fro many times, and we have trimmed it down, we've expanded it and so on, but what we have today is really an extension of something that has occurred every single year for 24 years in the department. We added in 2002 the Sachdev Memorial Lecture after the sad passing of Ved Sachdev. In 2007, Dr. Post and his family established the endowed Kalman D. Post Award. Actually, I think the title includes more than that now, for the best resident publication. And it was intended to provide an incentive and to recognize and to promote excellence in scholarship among the residents, with a panel composed of the professors in the department to judge the quality of those papers that were published. And so it seemed fitting to Cal and his family to do this, and just underscored his continued and ongoing academic scholarship that he has pursued to this very day. Today, we have a really fantastic lecturer to celebrate the 18th annual Ved P. Sachdev Memorial Lecture. I want to just tell you a little bit about Dr. Sachdev, who I had the enormous pleasure of working with when I was a junior attending. Ved completed residencies in ENT and neurosurgery in India before moving to England, where he became a fellow of the Royal College of Surgeons. He then came to United States and at the age of 39 began his neurosurgery residency at Mount Sinai under Sidney Gross, and subsequently became Lenny Malice's only real partner and the only person he ever really trusted to the extent that I saw. He worked here for 30 years, and when I arrived, he was a legend in his own time, full of grace and humor, knowledge. He, with his general and ENT surgery training, was a firm believer in neurosurgical airway management. And so he was really an intensivist before his time. He taught us how to do tracheostomies, C1-2 punctures, as well as a superlative intracranial and spinal surgeon. He also was a master storyteller, often seemingly rambling through many, many minutes, only to circle back and close with something that would leave you laughing for years to come. After his sad passing, his wife, Ronit, established and gave a significant amount of money to the department in memory of her husband to establish this lecture. And I know that both of them would be extraordinarily proud to see what their daughters are doing. Some of you may know, because Rivka was at Beth Israel for many years, both Rivka and Olka did their medical school training, went to medical school here. Rivka then went to SUNY Downstate, did her neurology training at Tufts, and then a movement disorders fellowship at Beth Israel with Dr. Bressman and team. She then stayed on for many, many years and has since moved to Cornell, where her career is thriving. Olga Sachdev also graduated from the Icahn School of Medicine. She's now associate professor of surgery. She did her vascular surgery training here, and as everyone knows, that is no small feat. She has won many awards. She is a prominent scholar/surgeon, and I know that both Wade and Ronit would be beyond proud to see what you both have done. So I do want to emphasize how grateful we are to Ronit and to the family for your generosity in establishing this many years ago. I want to apologize for not connecting with you earlier in the process this year. I can assure you that will never happen again. Over the years, this has become a cornerstone of our educational program. I want to share with you, in case you don't remember, some of the people who have given this lecture. All of them are prominent physicians and scientists, some fully in neurosurgery, some scientists, some in ENT. And as you can see, we've had very, very impressive individuals provide this lecture. So with that, I want to introduce this year's presenter, Dolores Hambardzumyan, PhD, MBA, recently recruited with the help of Kostas Hadjipanayas from Emory. She is professor of neurosurgery and oncological sciences with a joint primary appointment in our department. We consider her one of our very own. You can see her education with a PhD from the Armenian National Academy of Sciences, an MBA from Emory in leadership and healthcare, as well as numerous fellowships. Before coming here, she had established herself as a premier brain tumor researcher with evidence of other outside recognition in the form of multiple NIH grants and R01 support, which she brought with her. As you'll see, her focus on neuro-oncology was in pediatric medulloblastoma and high-grade adult tumors. And I won't read everything here because she's going to tell us more about this. But her move to Mount Sinai and establishing her lab marks a new era in the science of brain tumor research in our department, and it fits beautifully with the legacy that Wade Sachdev has left. So Dolores, it's a pleasure to welcome you. and we look forward to your lecture. I'm going to stop here, and this is where the slides will pick up after her lecture. So I'll stop sharing, and then we can turn it over to you. You're muted, Dolores. Can you unmute?
Oh, thank you so much for a very generous and kind introduction. And it's a great pleasure to be today's presenter. Let's see if my technology will. Do everybody sees my screen? There we go.
You go to presenter mode?
Yeah. Do you see my screen?
Sorry, now we also see the other slides too, so.
You see other slides.
You should click Use Slideshow on the top.
Okay. 1 second. Let's see. I close those. Slideshow. Let me see.
Go ahead and click on that button there.
I did.
And now, do you see near the top left of your screen something that says Use Slideshow?
Let me see. Unfortunately, I don't. Let me see.
All the way up top left.
Which
Right.
okay, it's on the bottom. And how about now?
Yes.
Done. Thank you. Okay, approve. Dr. Pedersen is okay, approve. Here we go. Okay, sorry for that. And now, but that's got stuck. Okay, perfect. Okay, I got it. Thank you, guys. Thank you for patience. So I want to start also my presentation. It's an unusual times, unprecedented times, and I'm a basic research scientist. It's been an incredible experience for me during this difficult time to log in some of your Zoom meetings at 7 a.m. and see how you were navigating and doing everything for patients, care, and staff. So it was very humbling for me to listen, and I felt like I wish I could do more. And I want to thank you all for your dedication, commitment, and sacrifices. And I want to thank very much Dr. Pedersen for his incredible leadership during this time. And it was great to see me from the side. So I joined the New Yorkers, thanking all of you. And by this, now I'm going to move on and start my talk. And it was supposed to be about my favorite topic, any topic in neuroscience. And I decided to focus on my favorite cells that I study, called myeloid cells, that are cells of your innate immune system in glioblastoma. I think this snapshot shows how heterogeneous, how intertumoral heterogeneity of this tumor and how difficult it is to study. And where do you start? And this is just a snapshot from Oncoscape that has all the genomics data on over 600 GBM patients. Where do we start this? How we study disease that's so complex and so heterogeneous? So over a decade ago, the Cancer Genome Atlas Initiative decided to sequence a large number of GBM samples. And what they did after sequencing, based on RNA expression, they cataloged these tumors into what now stands into 3 molecular subgroups, and called preneural, classical, and mesenchymal, after the similarity of the normal cells that they resemble. So this, by no means, those are mutually exclusive. And in contrary, there are lots of overlap, meaning you can have a preneural tumor that will have cells with a classical or mesenchymal signature. What that shows, though, that at the given time of tumor resection and location that was resected, preneural means that predominant transcriptional program of that tumor will be more preneural. It depends where you put the threshold. So majority of cells will have preneural signature. Later on, we learn also that there is a subset, which is probably the biggest discovery of Cancer Genome Atlas Initiative, that we have tumors that are IDH1 mutant and GCM, and those are the tumors where have a global hypermethylated phenotype, and the rest of the tumors are divided into IDH1 wild type. And this has a very important clinical relevance because tumors with IDH1 mutation, glioblastomas, live much longer. So, and again, very difficult to model RNA expression profile because you have thousands of genes up and down. But when you look at that, this is the heat map of genetic alterations that come with these expression profiles. And look at the copy number alterations and mutation heat map. One obvious thing is that glioblastomas really have a low mutational burden. But when you look, there are some common tumor suppressor losses like p53 loss, you have CDKN2A, which is very prominent across all the subtypes, you have PTEN loss. And when you look at closely signaling pathways associated, you can see that although not mutually exclusive, for example, PDGF receptor amplification more predominantly happens in a proneural subgroup. Then you have EGFR, including EGFR V3 mutation, which is the most common mutation in classical. And when you look at NF1 mutation here in this corner, you can see although you will have some NF1 mutant tumors in other subgroup, majority of them cluster in mesenchymal subgroup. So this is important because this is the information we use in the modeling human disease in mice, which is the strength of my laboratory. And then the same thing on RNA expression and also genomic alteration was done on a pediatric high-grade gliomas. For many, many years, we were thinking that pediatric tumors are so similar to adults. And any trial that was designed for adult gliomas was also used for pediatric tumors. But thanks to genomic initiatives, high-resolution genomic initiatives, we also learned that pediatric tumors are actually very different from adult gliomas. They share a common mutation with adults with this PDGF receptor amplification, which is the most common mutation in pediatric high-grade gliomas. But then you end up having specific locations, as you can see in a brainstem and in a hemisphere, that are associated with pediatric tumor-specific histone mutations. In this particular case, in contrast to adult gliomas, histone mutations lead to hypomethylated phenotype, and that's associated with worse clinical outcome. So meaning tumors, for example, that are formed in a brainstem, over 80% of them have either H3.1 or H3.3K27M mutation, which co-occurs with ACVR1 mutation. And those are the tumors that have the worst outcome. And then you have the hemispheric tumors, and around 7% of them will have H3.3G34RV mutation, and the majority of them will not have it, but will have CDKN2A or p53 mutation. The reason I introduced all this genetic alteration, you'll see in my next slides, this is the information that we use to accurately model using genetically engineered mouse models. So in order to be able to study immune function. But before I introduce the modeling system, it's all my favorite quote is from George E.P. Box, who is a biostatistician, that says all models are wrong, but some are useful. And hopefully I will convince you for the specific question of studying immune cell interaction with tumor cells. I think having immune competent murine models that closely resemble human disease is probably the better choice, although not the perfect choice. So how do we model genetics of human disease into the mice using genetically engineered mouse models, which I actually just by 2 slides introduced you major genetic alteration in human disease. Then we take these genes, we convert them into viruses, and we use viruses as delivery platforms to deliver the specific gene into the brain of mice. So most of genes do not cause cancer. There are secondary mutations, but you can have genes that will cause a cancer, and those are the ones that are driver mutations. And you'll hear across my talk referring to them as a driver mutations. So the viral system that we use, it's called RCAS TVA, was developed by Harold Farmas and first introduced for brain tumors by my postdoc advisor, neurosurgeon scientist Eric Holland. And that's based on chicken virus. Mammalians, we and mice, do not have receptor for the virus, and so we are not infectable with the virus. Because for virus, in order to get into the host cell and integrate into host genome, it needs a receptor. But we have genetically engineered mouse models that we drive expression of the viral receptor and the specific promoter. In this case, we use nestin, which labels stem progenitor population, and both adult and pediatric mice, baby mice. And you can use this system to deliver different oncogene and tumor suppressor combination to generate de novo primary GBM in mice. And that's when you use adult mice, and you can use mice from 4 to 16 weeks of age, basically, if you want to model different age categories. And you can use them in a newborn mice, and you can inject exact the location that you choose specific to pediatric and generate pediatric high-grade gliomas. So in this, Yeah, that's how we make, for example, I won't talk too much about pediatric tumors today. I'll talk about adults, but I'll just introduce how we use that information and generated variety of pediatric high-grade gliomas. So we use PDGF signaling, which is the most common mutation in pediatric. We inject in a hemisphere and we do hemisphere-specific histone mutation, and then we end up having tumors that this is an H&E section. You can see they recapitulate key histological features of glioblastoma. You can see pseudopalisating necrosis, microvascular proliferation, high Ki-67 index. Those are very highly proliferative tumors. And they are nicely, you can MRI them with either T1 or T2 imaging. And to model like brainstem gliomas that are called in clinic also diffuse midline gliomas with histone mutations, K3.1 or 3.3 K27M, we do the same combination and we end up having a location in a brainstem. This is the MRI images. And those are very invasive tumors, again, key structures, and you lose in these tumors when you use histone mutations, K27M-E3. So what we wanted also to do, okay, we use the genetics, we get the histological features and imaging properties of these tumors to be as close as we can to human disease. But for immune studies, we wanted to see whether a global immune profile of these tumors we can recapitulate using the same genetic alterations. And this is, we did use the archived FFPE sections of around probably 20, 30 pediatric high-grade gliomas. Those are much rarer than adults. So, and we did 770 immune-related gene-specific RNA sequencing. And then we clustered them in unsupervised hierarchical clustering. As you can see that tumors, diffuse midline gliomas clustered together pretty closely, and hemispheric tumors clustered together. But I want this heat map to see that higher red color means higher inflammatory score. And it's clear that diffuse midline gliomas have higher inflammatory score than hemispheric tumors. So before we do any studies, we wanted to ensure that we can recapitulate global immune inflammatory score that we see in human patients in murine model. And this is tumors generated with histone mutations, the DIPGs, and hemispheric tumors that are histone wild type. And then when we do unsupervised hierarchical clustering, you can clearly see these guys, DIPGs clustered together, and their inflammatory score is as high as you can see in a human patient sample. So this gave us a confidence that models are useful at least for immune studies. Similarly, when we target neural stem progenitor cells in adult mice using stereotactic injections, where we can deliver exact location where we want to introduce our viruses, we can generate signatures that are proneural corresponding to human. There we use a major oncogenic driver, specific for proneural PDGF signaling in combination with short hairpin to silence p53 gene. We can use the same thing if we use NF1 and short hairpin p53, so we silence both tumors. Hello? Oh. And we can generate mesenchymal tumors. And if we use human-specific RE3 mutation, and this is a very complex mouse. With CRE, we can delete P10 and we can induce RE3 expression. We get tumors that resemble very closely classical tumors in human. So those are the survival curves. I think murine models' advantage also in some ways is that you can see differences in survival curves, which usually you don't see in human case, because in human, you get the patient with tumor already fully blown and formed. In mice, it's a process that represents initiation of tumors and then progression. So this difference in survival seen, we showed is due to initiation, meaning the transforming ability of these oncogenes. But the moment they develop, there's no difference in the growth genetics. And what we end up having with those, we have majority of them are glioblastoma, grade 4, and now we have grade 3 anaplastic astrocytoma. And when we look at the histological features, here it's a contrast enhancement in human GBM. You can see this is H&E of human GBM, beautiful pseudopalisating structure, and you have microvascular proliferation. And when you take the murine GBM, it also enhances with gadolinium and MRI, and you can see beautiful structures preserved. You see pseudopalisating structures and microvascular proliferation there too. What we did when we did the similarity matrix to see how different from one another are murine GBM, you can see that PDGF tumors cluster together and F1-driven tumors cluster together and EGFRs cluster together. So although there is lots of similarities, but there are differences that are sufficient for them to cluster together. When we took a panel of 30 genes and we ran qPCR with human known genotypes versus the murine tumors, you can see, for example, PDGF-driven murine tumors cluster very closely in this PCA clones with the preneural human GBM. So they cluster closer than preneural GBM, for example, with the classical human GBM. So that's close enough for us to consider that the modeling is probably sufficient to move to the next step. Then we did the 770 gene analysis again and wanted to see if we see differences in an immune profile in the murine models. And you can see that PDGF tumors cluster closely together. And then you have EGFR and NF1s have the highest inflammatory score. And this is it's aligned with the published literature showing that mesenchymal tumors actually have higher inflammatory signature and more macrophage infiltration. So we saw the same things in murine tumors. And with this, we were confident that the models at least are closely resembling the disease we are trying to model. We decided to study cells of tumor microenvironment. So tumors are, if you take a piece of the tumor and dissociate, the question was for us, this is just the work of many years, what is the composition percent-wise tumor cells versus the non-tumor cells that form tumor microenvironment? And in human, there are several cell types that we don't have surface markers that we can pull down and get a right quantitative analysis there. But in the murine models, we have lots of reporter mice that we can use and combine with the FACS analysis for multi-panels. And what we discovered there that around 60 to 65% of this tumor that we resect, this is a bloody GBM from mouse, only 60 to 65% of that mass actually consists of real tumor cells. The rest, 35 to 40%, is non-neoplastic cells of tumor microenvironment that come to the tumor and contribute to tumor growth. When put quantitatively, the most abundant immune infiltrates in GBM are the myeloid cells, and they are mostly referred in literature as tumor-associated macrophages. What are the tumor-associated macrophages? So before I go and start dissecting further what are those cells and what do they do in glioblastoma growth, I wanted you to introduce what we have in a normal circumstances. We have a healthy mouse, we have a CNS. We have our own myeloid, CNS-specific myeloid population that's called microglia. Those are your resident myeloid cells. And they form very early during development from your exact progenitors, and they come to CNS and they reside there. And they stay there forever. They can live forever. They can self-renew like stem cells. And independent of what kind of injury you have, they get activated. When they get activated, they change their expression profile. They are genetically stable, but they are very plastic cells. And depending which kind of injury to CNS you have, their activation can be very different. In another hand, in a parallel universe outside of CNS, you have inflammatory monocytes. We don't have macrophages in the blood. Those macrophages are in general stationary cells, so you don't see them in circulation. But what we have in blood circulation, we have monocytes. Those are progenitors of macrophages. We have two subsets of monocytes. We have inflammatory monocytes and we have circulating monocytes. And those guys are not inflammatory in general, but they are called inflammatory because those are the cells that migrate towards inflammatory side when there is an inflammation. If there is no inflammation, these cells cannot self-renew and cannot live forever. In fact, they live only up to 48 hours. And they're constantly replaced by hematopoietic stem cells through stages in bone marrow until you get a monocytes that exits bone marrow in circulation. And it's a constant process that hematopoietic stem cells provide into circulation, new inflammatory monocytes. So to start studying tumor-associated macrophages, we took a bunch of archived, again, human adult glioblastoma samples, and we did this with collaboration with Dan Pratt at Emory. He provided us around 50 samples that we took the FFPE sections, did an RNA expression profile on those, and then assigned them based on the signatures. Are they preneural, mesenchymal, and classical? And did a basic experiment of staining them with IBA1, which is a pan-macrophage marker. So it basically labels all the macrophages, monocytes from circulation, every myeloid cells, but neutrophils. So when we did and we quantified, you can see that there is a difference. When you look at mesenchymal tumor, similar to what was published in literature, those cells are enriched by macrophages. They almost have twice more macrophage-occupied area compared to classical and preneural tumors. And when we did the same exact experiment using our mouse models that closely resemble the subtypes PDGF, NF1, and EGFR-V3-driven tumors, you can see we see exactly the same signature. So these tumors have twice more macrophages. That raises the question, why? And what do these cells do in these tumors? But when we took AIF1, which is the gene that codes the protein IBA1, and did then, there's a perfect correlation there between gene expression and protein expression. And we took within the subtypes, put the high and low expressing, and did the correlation with survival. In preneural, high IBA1, IIF1, samples with high IF1 expression had a worse survival when it was opposite in mesenchymal, which was very intriguing for us to see. That's probably not the number of the cells, it's probably their profile and composition that dictates in a given GBM what is their function. With this, we went on to immunophenotype using a FACS panel, and I won't go into great detail of multiple 15, 14 color FACS that we use to define its cell population, but you can see in this plot, red color is going to be microglia. It's a resident myeloid cells. BMDM, which is going to be bone marrow-derived monocytes and monocytes-derived macrophages, and neutrophils. And it's interesting that neutrophils are more prominent in circulation. They are 7-fold higher than monocytes are, but you see a reverse in a tumor. You see very small neutrophil infiltration and very large inflammatory monocytes infiltration. With this, we used this panel and characterized the immunophenotype myeloid population in these tumors. And you can see in mesenchymal, although we have twice more prominent population of myeloid cells, it's actually resident microglia, whereas in PDGF and EGFR-V3 tumors, you have BMDMs. So those are all cells that comes with the millions from circulation. So of course, it's very important also to understand where are these cells localized anatomically in the tumor. And this is a reporter mice that we use to characterize their location. And CCR2, which is in a red color, is specific for inflammatory monocytes. It's not expressed by resident myeloid cells. And you can see this is the tumor border. You can see in the peritumoral areas, we have only green, single green positive cells, and those are microglial cells. Where inside of the tumor, around the vasculature, which is in the purple color here, you can see all the cells from circulation. So they come and they reside in the perivascular areas. So the question for us was, why in the perivascular areas? Why they didn't migrate further in tissue parenchyma, and how early they come from circulation. And this is just, we took tumors from various sizes during development, from the smallest tumor that you can't even see visually to the going in the scale into larger ones. And you can see with the red is all from circulation, red and yellow, everything comes from blood. As you can see, even in the smallest tumor, the cells are coming. So they come very early during tumor development. And when we did ratio, yellow is our cells from blood and GFP single positive cells are our resident cells, you can see pretty much there's no statistically significant difference between tumor size and ratio of these two populations. To better understand their location in perivascular areas, because those are crucial areas that are most resistant to radiation therapy, and to understand the dynamic and really trace these cells from circulation to the brain, we used multi-photon imaging that would allow us to generate tumors in mice, then do a surgery and take off the skull and put a glass, and you can image this mice live and label the different population cells that we are interested in with green, red, and then follow their dynamic of infiltration over the days and weeks. And the technology also can be used to evaluate immediate effect of therapy on both tumor cells and tumor microenvironment. And you can see this is how it looks like when we cut the window and we glue it. And this is the vasculature we visualize by injecting red dye dextran. And this is just the coronal section of mouse brain, and you can see how GFP positive green cells are your resident cells. We don't have cells in a normal brain from circulation, and they are uniformly distributed in here. And what happens, they are about, based on the literature, you read 10 to 13% of total cells in CNS. And we did start tracing these cells to see what's the dynamic of their movement, and if you can see what happens to these cells. They constantly extend and retract their processes, but their body here doesn't move. And we did this for hours and days. So macrophages are really stationary cells. So but when we look at the monocytes, this is the monocytes, and they are labeled green in circulation in a reconstituted mouse. And those are small round cells, very dynamic progenitors. And what happens when you have, this is the tumor vasculature, and you can see tumors are black, so you can't see them, and you only can see the blood vessel red, and you can see how this guy is crawling out. So what happens later? And so we did this for longer hours to trace what's happened to that cells that gets out there. And this is the bone marrow chimera reconstitution, so I won't go into great details. But because of the infiltration of these cells so high, we had to dilute monocytes to be 50% of cells that we could trace them from circulation to go. So we could do that like this. We could have only 50% of BMDM and GBM to be green, or we can do reverse experiment where our bone marrow derived cells are, and we can visualize that they are black and microglia is green. So we could study dynamics of both cell types. And this is a microglia, and this is in a normal brain. This is what happens when you have a tumor. You can see the density is much less in the tumor. Instead, you see them accumulated in a peritumoral areas. And what happens in the tumor, this is the imaging of microglia within the tumor. And you can see they still extend and retract their processes, but they don't move. This is probably one of the reasons they get pushed out and accumulated in peritumoral areas. While when you look at the monocytes, and this is a naive brain, DAPI staining, you don't have monocytes. But when you have a tumor, GBM, you can see how abundant those cells are from circulation in the tumor. And then when you image them, you can see small cells getting out of the vasculature. But the moment they differentiate, and they become large macrophages, they also, like microglia, extend and retract their processes, and they don't move. And this process we calculated is extremely fast. This is why the moment they get out of the vasculature and move just a slight distance from the vasculature, they differentiate, and that's why they get accumulated in the perivascular areas. And we could characterize them by different technology, show that those cells are major immune infiltrates. We also could, when they differentiate in a 5 micron sections in FFPE, usually cannot distinguish them when they become macrophage, but with thick sections and 3D reconstitution, you can clearly see there is a morphological differences in the cells. And we could trace for hours the movement of each cell and show that really these cells are dynamic and they move. But once they differentiate, they actually upregulate the GFP expression. So there is a reverse correlation. Lower GFP, more dynamic, and that's your monocytes. And when they differentiate, they upregulate GFP expression, and they become more stationary, so they don't move. And this is just to show that microglia move even if we image them for like 3 hours. And then we used, so for us, those are very tumor promoting, and I will show some survival curves so you will follow me. Why is it important to study the cells? So we wanted to understand what is it that driving the cells come from circulation in this such a large numbers. So is it the compromised brain-blood barrier in glioblastoma, or it's, but that's not a physical holdout. Any cell can get in because we only specifically have infiltration of these cells. Or it's a chemokine gradient because the cells usually migrate towards chemokine gradient where it's increased in inflammatory sign. So to answer this question, we took both approaches. First approach was to normalize vasculature using bevacizumab, which is anti-VEGF antibody, and see if the vasculature is normalized and less leaky, we'll be able to decrease the infiltration of the cells. To do that, we generated tumors in mice, and then we treated them with anti-VEGF antibodies, which is bevacizumab's murine analog from Genentech. And then we put a cranial window and continued treatment and observing the cells. When we quantify it, you can see shape is different because we decrease the infiltration of the cells and all we have is differentiated cells. So we did decrease, but we didn't completely eliminate their infiltration. We barely made it significant, and that's translated for significant survival advantage, but not impressive. So the question was, yes, the answer is vascular leakiness probably contributes to infiltration for the cells, but that's not the only reason the cells are migrating to GPM. So to look at the chemokine and see what's the chemokine gradient role there, we focused on MCP family, which is called monocytes, chemoattractant proteins that are responsible for infiltration of the monocytes from circulation to the tumor site. Sorry for my slide. And it has 4 members in a mouse and human. And then it's interesting because they're all localized next to each other in mouse chromosome 11 and in human chromosome 17. And one of the biggest problem of this is that there is a redundancy in function. This is one way why all the chemokine, single chemokine targeted therapies so far were not effective. You target CCL2, CCL7 will take the function of CCL2. Many of them share receptor. So to overcome, and the most studied one is CCR2, basically leading their directional motion. To overcome this caveat, though, and we did it, we did GPM in a single chemokine knockout mice, and we saw the redundancy of it and didn't succeed it. So we had to take 2 approaches, parallel approaches. We had to CRISPR-Cas9, do editing and delete entire this segment on chromosome 11 in a mouse. And we made a mouse that doesn't have all 5 members of this family. And at the same time, we're looking at the switch. We were looking if there is any cytokine that is responsible to upregulation of expression of monocytes, chemoattractant protein family that we can target from the top and decrease the level of all members of the family. And we identified IL-1 beta being one of it. And when we did IL-1 beta expression, high and low in preneural GPM, because this is the PDGF B-driven mouse model that we used, you can see that samples, patients with higher expression of IL-1, it's here red line, have significantly shorter survival compared to the black line that represents patients with lower expression of IL-1. And when we did the same experiments in mice, you can see we recapitulate the same picture. So now we wanted to see whether in IL-1 beta knockout mice, we have decreased monocytes infiltration, which is responsible for this extended survival. And you can see that, how clearly we can show that. But that was a genetic approach, and then the beauty of genetic approach is 100% gene is gone. It's very difficult pharmacologically with drugs to achieve that effect for many reasons. So we did also look, because IL-1 signaling has FDA approved that anakinra that is used for many inflammatory diseases, and many of you probably heard about anakinra trial for COVID-19 patients with the cytokine storm. And we did the same experiments. We generated tumors, but in this case, instead of genetic, we took a pharmacological approach. When tumors developed, and because this is a peptide, recombinant peptide, to ensure that we deliver in the tumor and bypass the problems with brain-blood barrier, we implanted guided cannula. So it has a top screw on top. So every day you can take out and inject your anakinra. And we also added a dye that could show us how much diffusion we get. I mean, we get fairly enough diffusion, not complete, but even with that, we get a significant extension of survival of tumor-bearing mice, both with anakinra, which blocks IL-1 receptor, or anti-IL-1 targeting antibodies when it's directly delivered to the tumor. So this basically shows one of the examples, one of many examples, how macrophages form this vicious loop with tumor cells. So you have tumor cells and you have macrophages. So what tumor cells do, they express high level of chemokines, and I didn't show that data for sake of time, but tumors are very smart. They express high level of MCP family chemokines with the goal of recruiting these cells come to the tumor. And when these cells come, they produce high level IL-1, which induces both tumor cell growth and also increases MCP chemokine expression to bring more monocytes into the tumor mass. And that's how you end up them forming this vicious loop of supporting each other growth. And just by my talk, I introduced that targeting therapy can provide extended survival in mice. This, if I return back to my introductory slide there about microglia and monocytes living in parallel universes in a normal healthy mouse, they meet actually in GBM. They come due to, hopefully I convinced you that vascular leakiness is a contributing factor for these cells exiting blood circulation and getting into GBM and differentiating into macrophage, and also chemokine gradient. I think the strategy should be targeting both at the same time, and we're working on it now. And this just to summarize what these cells, besides promoting tumor growth, they do. Those are all the factors published in literature that will responsible that TAMs would secrete to increase tumor cell growth. And those are all the factors that are released by tumor cells, people showed in different GBM models that are chemoattractant for TAMs to come to the tumor. Those are the outcomes. So microglia in invasive age promotes tumor invasion. You have myeloid cells from circulation promoting tumor growth. You have immunosuppression, and now we basically don't have much adaptive immune cells, T cells coming, and probably one of the reasons immunotherapies are failing in GBM because we don't have these cells coming. And these cells, myeloid cells, are also immunosuppressive. They can suppress the function, and there are studies showing it. And recently, we also showed that myeloid cells, since they are in a perivascular areas, we didn't know about their function and involvement in tumor-associated vasogenic edema. With this, I want to thank, and I think timing is good. I will give you time for a few minutes. I was also asked to do the career advice for academic career. I mean, I can by no means give a career advice for neurosurgeons. I can only admire your job, but I can tell you on my personal experience what helped me to go through it. So this is my team members basically did all the work. They were extremely strong individual contributors. This is my team at Emory, and we just moved and we got the shutdown. So new people that I hired weren't able to join my lab, but James and Cameron were graduate students, absolutely brilliant graduate student. Now they graduated right before I moved to Mount Sinai, and they are postdoctoral fellows, both looking at immunotherapy in pancreatic cancer and in GBM. And Ben was an undergrad that worked at nights for 2 years, got basically in every medical school applied, and Zio has been instrumental driving the science in my lab, moved with me, and is currently here at Mount Sinai. But despite the success in a small setting, and I have always had a smaller lab because I wanted to have enough time to contribute to everybody and be available for everybody anytime they need it, I think the success of their work was not only because they were a very good individual contributor, but because they also worked very well as a team together. And just a few words, I have time, 5 minutes. I'll just give you a brief introduction, what I learned on my experience on academic career, what to do. First, I want to just say I have an immense passion for science. And I think the key to be successful in science, research, and anything probably if you have a passion for it, it's a biggest driver. So this is my development in academic career. It started, finished my PhD in 2002 and started my postdoc in 2003. And then it went through all the steps. I think the key as a postdoctoral fellow that I now look back, I mean, I was born in Armenia and I got my education there. And I moved for a postdoc pretty much in 3 different countries and 2 foreign languages. So it was pretty, not an easy journey, but I think the key messages that helped me to make it work, make it happen was probably adaptation, because cultural and language, and also my mother always, even when I was a kid at school, would tell me that persistent hard work and dedication, pretty much you'll overcome every difficulty you have and you will learn and survive. And this is just pretty much my postdoc journey. I did a couple of months training in UK for MRI to learn how to MRI mice. And then I need a year of postdoc in France, and most of my major postdoc, 6.5 years, was in New York City in Sloan Kettering. And then I started my laboratory at Cleveland Clinic, and that's in 2010, 2015. And I think I've learned that's very different from being individual contributor as a fellow. So you were put in a leadership position running a lab, which involved different set of skills. And that's where I felt like lots of improvement I could use and learn. And 2015 to 19, I was at Emory University and recently moved to Mount Sinai. At Emory, I hired 2 graduate students, and for me it was very clear it's a very big responsibility because it's in the beginning of their career, and I wanted to be better prepared, and I could see some weaknesses that I could use to learn. So I did an MBA, which was an unusual step, and every Friday, Saturday, Sunday, I would sit and learn about leadership. And what I learned to be effective in academic career, be the head of the lab or any other leadership, when you have to manage more than one person or even one person, you have to learn to manage up, down, and sideways very well. And you need to be equally good at managing all 3 of them. What I also learned, which I can put in very easy, simple graph, it's a corporate word. In a corporate world, from individual contributor to become a business unit manager, you really have to go through multiple stages, and it's a stair and evaluation, training you for a different set of skills. What happens in academia, from individual contributor, from postdoctoral fellow, you jump up like an elevator into business unit manager without sufficient training in leadership and management. So that I think, and that when that happens, you have to be very successful at getting an NIH funding, publishing manuscript, teaching, and building obviously functional lab. There, I think we were prepared to do all 3 of them. Of course, you as an individual contributor, postdoc, you always have to think you need to publish papers to get NIH funding. You can learn to get NIH funding. And one advice I have, which worked for me, was to serve in NIH study sections, to work and learn what makes the grant and what breaks the grant. And actually, when you are present there and doing it, it's the best learning experience. It's an extra work, it's an extra hard work with weeks and weeks on reviewing grants, but I think that's the key for learning how to do NIH funding. And teaching, obviously, also very important, because by teaching, you can select some students to come to your lab better. But in the red, I highlighted where I had the struggle the most, and I think I would advise everybody to take any leadership courses of management and stuff that you can when you start your laboratory as an independent. And now you are also practicing neurosurgeons, and I don't know, I barely manage this part of the work, but Kostas and Jenny Zhao, they can tell you better how they managed to be a practicing neurosurgeons and doing all of this successfully together. But so what I learned, and what key message is probably here, it's the most important thing is the talent management, and that's the human capital. It's very important.
1 minute warning.
Oh, perfect, and I'm done pretty much. It's recruitment, replacement, and then knowing when to promote and when to select. And the key aspects I learned and I used, and I could see directly effect on my graduate student, that's giving the candid feedback, making sure that the goals, personal goals and lab mission are aligned, then you both are on the same page, and outline clearly expectation, and have a bit more affiliative on coaching style in management. Usually works much better on people. And with this, I want to thank everybody, and I hope I didn't rush too fast, and I'm happy to take questions. And Dr. Pedersen, I'm 4 minutes earlier, 5 minutes.
Well, thank you. That was just, it would be very hard to beat expectations, but you beat expectations.
Thank you.
JV, do we have time for some questions?
Unfortunately, we all, what I'm going to do is read a question from Ulka, who joined us, and then we're not going to have time for the response, but I'm going to ask Dolores
To type the response.
to type the responses as part of the Q&A, because we have such a tight schedule. So you'll see there that there are questions for you to answer, and Kostas, if you can do that, then the whole group will see your questions as well. You know, you like to take at least one thing away from every talk, and although I am not as facile with the brain tumors as I would like to be, I definitely related to this slide here. I'm going to remember this one, that all models are wrong, but some are useful. I looked up who this fellow was, and sure enough, he's a statistician, was a statistician. So I'm sure that Jay Mako, who's also a statistician, can relate heavily to this. So thank you very much, Dolores. It was really spectacular as expected, and we look forward to hearing more from you as well as the questions that people are asking. But we're going to go on now to Research Day presentations. Here are the presenters that you can see here. And I want to thank the faculty sponsors who, without whom this research would not be possible. And you will see them reflected in the talks. This is the timeline. And as you can see, it's pretty precisely controlled. And so with that, and then at the end, Dr. Mako will present the awards for the post and research award. So with that, I will stop sharing and turn it right back to Chris.
Okay. There we go. Just making sure you can hear me. All right, so we are on a very tight schedule, and we're going to move through presentations. We're going to have 7 minutes for each presenter, and then 3 minutes of questions. And I'm going to give a 1-minute warning. And at 7 minutes, I'm going to have to stop the screen share. So I'd really appreciate it if you stick with it. And we'll have time for questions and 1 minute transition between speakers. So we're going to start with Alex Schuper, and the sponsor is Costas. We're also going to make sure that the sponsor is present. So we will be messaging sponsors, and we would appreciate if the sponsor were available and on the talk before to make sure that we know that your tech is all set. So Alex Schuper, are you on and can you screen share?
Yes. Can you hear me?
Yes.
Okay, great. You can see my screen okay?
Yes.
Okay, great. So good morning, everyone. My name is Alexander Schuper. As Dr. Kellner mentioned, I'm a first-year neurosurgery resident here at Mount Sinai, and today I will be presenting our group's work entitled 5-amylolevulinic acid for enhanced surgical visualization of high-grade gliomas, a prospective multi-center study. I personally have no disclosures, as well as the PI of the study, who's Dr. Young. Dr. Hambardzumyan is a consultant, excuse me, for NXT DC, who makes gliolin. So just to start with some background, starting at Stoop and multiple studies since have redemonstrated that greater extent of resection is associated with longer survival in patients with high-grade gliomas. With the use of fluorophores, such as 5-ALA, extent of resection, safe maximal extent of resection can be improved by enhancing intraoperative visualization of these high-grade glioma tissues. 5-ALA is an oral solution, and upon ingestion is converted into protoporphyrin 9, which is in the heme synthesis pathway, and it subsequently accumulates intracellularly in glioma cells, which upon excitation by blue light fluoresces, as seen in the video below. It fluoresces in a red to violet color upon surgical resection. In 2006, Walter Stumer et al. Conducted a phase 3 randomized controlled trial, which showed that at a 6-month time period after use of 5-ALA in glioma surgery, there was improved 6-month progression-free survival compared to controls who underwent resection with white light. Since then, 5-ALA has been used very widely across Europe. However, as with other things in medicine, the US often lags several years behind. And with large thanks to Dr. Hambardzumyan, recently in 2017, 5-ALA received FDA approval for use of visualization of glioma tissue during surgery. Since then, there have been several single-center studies, though to date there have been no multi-center trials assessing the use and safety of 5-ALA in glioma surgery. So our primary objectives for this study were to determine the diagnostic accuracy of using 5-ALA upon glioma resection with regard to intraoperative fluorescence, and as well as to assess the safety of 5-ALA use. Similar to other studies looking at glioma resection, we did an exploratory post-hoc analysis to evaluate the extent of resection achieved in our cohort. As far as our methodology, we performed a single-arm prospective multi-center study using 16 centers, including our own here at Mount Sinai. We included all adult patients between the ages of 18 to 80 with KPS scores greater than 60 upon baseline. And they had MRI diagnoses of newer recurrent surgically resectable high-grade gliomas, which were defined as WHO grade 3 or 4 tumors. Each patient received between 3 and 5 intraoperative tumor samples, and the fluorescence status of each tumor sample was recorded. After surgery, patients were followed for 6 weeks to track adverse events, and as well as changes in their neurological exam. And then, as I previously mentioned, we performed a volumetric analysis on pre and postoperative MRI imaging. Our basic results are shown in the table. We had 225 samples, of which the vast majority were high-grade gliomas. As we saw, as you can see below, there was a very high sensitivity for high-grade gliomas with over 94%, as well as a positive predictive value of 99%. And intraoperative fluorescence was fairly accurate, with an accuracy rate of over 93%. There were several patients who had adverse events. 2 patients experienced a skin rash following administration, and 1 patient developed a transient fever postoperatively that was felt to be potentially related to 5-ALA. Additionally, two patients in our cohort developed postoperative meningitis. 1 patient had a neurological decline secondary to tumor progression, and there were 2 postoperative deaths in our cohort. When we tracked KPS status, there were no significant changes between baseline, the day of surgery, and up to 6 weeks out following surgery. In our post-hoc analysis, we had mean extended resection of 88%, with mean residual tumor volume of just under 3 centimeters cubed, and a gross total resection rate of 64%. This is comparable to rates that have previously been published in the literature, including a recent meta-analysis looking at both 5-ALA and white light resections. So in conclusion, 5-ALA was well tolerated within our cohort. The use of 5-ALA was not associated with significant declines in KPS when patients were measured up to 6 weeks postoperatively. We found that the use of proporphyrin IX fluorescence from 5-ALA was fairly accurate, and our numbers corresponded well with reported values in the literature. The major limitation of this study is that by being a single-arm study, we have limited ability to make conclusions on extended resection, and we would need a stronger control sample to make more definitive conclusions about the effect of 5-ALA on extended resection. I would just like to thank my mentors, Dr. Young, Dr. Hadjipanayas, and Dr. Germano, as well as our team, Rebecca, Will, Jessica, and Jan at Sinai, as well as our collaborators from other institutions. I'm happy to take any questions.
Hostess, any comments?
Yeah, no, so I'd like Ray to say a few things too, but great job, Alex. That was well presented. And I think, this was a large effort that went over multiple years, and Isabel helped with this as well initially. And I think, it's nice to see that our study results really kind of came in line with other studies in the randomized controlled trial that occurred. This is basically the only multi-center study ever performed in the US with 5-ALA. So I'd like to thank also Ray Young, who's really helped with this.
Yeah, it's, I'd just like to comment also that, I thought this was a very good, demonstration or confirmation of, some of the sensitivity data that's out there, basically confirming that the positive predictive value of 5-ALA is quite good, even in sort of real world conditions of resection, which really was this study, in that there were so many different centers involved and kind of different settings for glioma surgery that were included, but yet the PVV was quite high, indicating that the fluorescence does really correlate with high-grade glioma pathology. And of course, the negative predictive value being lower is also in keeping with what's been reported. And really does depend a lot on how the sampling algorithm occurs in some of these studies. So, that tends to fluctuate a lot. And then just to re-emphasize the point at the end that Alex made about extended resection, it is very hard to interpret in many of these studies. There's a very wide range for any type of adjunct or no adjunct used in surgery. So certainly more research will need to be done to kind of clarify the actual benefits gained by 5-ALA.
And this is Jay. Just I would just mention to Alex for your contribution and Ray and Costas and Isabel, this is to run a multi-center study like this is no small feat. It takes a tremendous amount of work. Having done it myself, it's a major effort, and you guys should all be congratulated. It's a big deal. So,
and you know, we also need to recognize our clinical trial team at Sinai. And Jay, you know this very well, how hard they work with the vascular studies, but they were a key part of the success of this study. So thank you, Ashley. Thank you, Jessica, not with us anymore.
This is Isabel, and I would like also to congratulate Alex. I think that he contributed to this last analysis. I think that the slides went by beautifully and very smoothly, albeit fast. And I just want to be sure that the audience doesn't get confused on the purpose of the study. The purpose of the study was not to assess 5-ALA to guide the surgeon resection. At that point, the 5-ALA was still not FDA approved when the study was proposed to the FDA. And therefore, the goal of the study was really to see whether or not 5-ALA had an histopathological correlate, which, as you saw from the slides, it does. So the extensive resection was not one of the aim of the study, number one. Number two, we did not keep track of who used it and who did not use it. So the data shown has to be taken with those considerations, as Ray Young did imply. So from our study, we really cannot conclude whether or not 5-ALA is helping the surgeons with the resection. And naturally, we all know that other studies were focused on that, and we all know that using 5-ALA is useful and helpful. Thank you all for your attention.
We also have a question from Corey Gill. Does IDH status impact fluorescence?
I can answer that. So Corey, good question. Unfortunately, there hasn't been any correlation found. There's been a few studies looking at that, but no, that hasn't panned out.
And I think in general, it would correlate with high-grade pathology, which, certainly IDH mutant tumors can have high-grade pathology as well as low-grade. So, there were, I think, some IDH mutant higher grade gliomas that were included in our cohort.
We have one more minute, and we have a question. We have somebody raising their hand in the attendees. Altaf Ali, do you have a question? Looks like you're muted there. Let me just explain the hand raising. There we go.
Yeah, can you hear me?
Yes, we can hear you.
So I'm a neurosurgeon. Thank you very much. It was a really nice presentation, and the team is an excellent one. I will just ask a brief question. Did 5-ALA affect post-operative imaging?
No, it didn't really have any tangible effect in terms of enhancement of tumor post-operatively. We took the contrast enhancement post-operatively to signify residual tumor or lack of residual tumor. Did I answer your question?
Thank you.
Okay, thank you. We're going to move on to Kurt Yeager. Kurt, are you able to share your screen?
Yep, pulling it up.
Well, Kurt's pulling that up. Let me just explain that if anybody in the attendee section, which is probably most people here, want to ask a question, just raise your hand, and then I'll give you the ability to speak, or you can type your question in the Q&A box, and that also works well. Okay, Kurt, go ahead.
All right. So my name is Kurt Yeager. I'm a PGY5 at Mount Sinai Hospital, Department of Neurosurgery, and be presenting today on the technical and clinical outcomes after endovascular thrombectomy for large vessel occlusion during COVID-19 era. I hope no one is experiencing burnout from the COVID-19 papers and studies yet, but we'll be presenting this. And I hope you guys remember back to a time when there weren't all these things like masks and gloves and gowns. This was the last time I think I had a mask, or I did not have a mask on my face in New York City. And then contrast this to March 28th when Dr. Shigematsu and myself were doing multiple strokes a day. And we've studied at Mount Sinai that in our unpublished results, there have been a significant burden of large vessel occlusions in New York City during the COVID-19 period. And looking at the timeline, we experienced this in live time, but there are some key points that we need to point out. The first case was of COVID-19 in New York was in March 1st. New York State determined a lockdown period, or determined a state of an emergency on March 7th, and then March 20th was the lockdown, the strict lockdown enforcement, quarantine and social distancing enforced. So what we wanted to really see is we wanted to see what the actual practical results were from the thrombectomy, comparing these COVID times to pre-COVID times. And so a lot has been looked at in terms of the relationship between COVID and stroke. And some groups have published on the increased rates of stroke. Some groups from NYU, for example, have found a decreased incidence. In our institution here, across multiple institutions, we've found an increased incidence of large vessel occlusion. And the hypothesis between the hypothesized link between COVID-19 and stroke come from really changes to Virchow's triad, as we learned in medical school. There's three main parts to that triad: the endothelial injury, stasis, and a hypercoagulable state. And COVID-19, the virus itself, can affect these in many ways. For example, direct viral invasion, having a lot of intravascular catheters in place, especially in the ICU, and then really a significant cytokine storming can cause endothelial injury, which leads to a hypercoagulable state. Stasis of the blood, for example, being in the ICU or being in bed for a while, can cause a hypercoagulable state. And then the medical side of things, an increased rate or increased amount of factor VIII, fibrinogen, and then just a general hyperviscosity of the blood is caused by the virus. These have all been studied and found to be true, and theorized as to why it may cause increased incidence of PE and stroke. So again, to kind of summarize our methods, we wanted to look at the effect of lockdown restrictions and social distancing on our workflow, the thrombectomy workflow and the preoperative workflow, as well as the practical outcomes doing thrombectomy, where the clinical outcomes the same or different compared to the pre-COVID times. And to do that, we took this block of time and compared it to a full year previous to that time. So or more than a year. We looked from January 1st, 2019 and May 4th to March 19th, 2020 as the pre-COVID time. And we assessed retrospectively our database of thrombectomy cases. We wanted to look at the workflow and outcomes. So the workflow, including the last known well to endovascular consultation time, that consultation time to the time when we actually make access to the vasculature, and then the access time to reperfusion time. Those are kind of three broad metrics that we looked at to indicate, number one, how fast people are coming to medical attention and how fast we're able to be notified. Then the time of activation to the actual transfer and to the patient getting to the angio suite, and then the actual procedural time itself. Our hypothesis was that everything leading up to the procedure may have taken a longer period of time, but then the access time to reperfusion, the procedural time, would probably be about the same. And looking at the clinical outcomes, we wanted to assess the endovascular reperfusion rates, so the ultimate positive reperfusion between the pre and during COVID times, and then the ultimate clinical outcomes, the change in the United Stroke Scale from pre-op to discharge is a short-term outcomes, just because we didn't have a lot of time to study the COVID group, and then the discharge location, whether they went home or went to acute rehab or something else. So in looking at the results, I mentioned the two periods of time. The pre-COVID time was January 1st, 19 to March 19th, which was just as the lockdown enforcements were being enforced. And then the COVID time was March 20th to May 14th, when we really pulled the data for this trial. In the pre-COVID times, we had 249 thrombectomies we performed, and then in the COVID period, there were 32. And so I'll go through the most significant variables here that we found to be different. And so the COVID patients were significantly younger, 65 years versus 71 years. Significantly fewer patients had comorbidity such as atrial fibrillation. So that was 3% in COVID patients, COVID era patients, compared to 30% in pre-COVID. And then the baseline MRS was significantly better in patients presenting during COVID times, indicating a better functional baseline. The presenting hospital also was significantly different. So people presenting to Manhattan were significantly decreased compared to those presenting in Brooklyn and Queens, which corresponds to the boroughs of highest COVID incidence, which is interesting. And then the baseline NIH stroke scale and the LVO location were unchanged, suggesting that the actual strokes were about the same. So looking at the workflow, overall, none of the times of the workflow were significantly different. The overall last and well to interventional consultation time, the consultation time to access time, and the ultimate reperfusion time were approximately, they were statistically insignificant. So could not make comment on those, interestingly. However, the clinical outcomes, the reperfusion rates were approximately the same, 91% in pre-COVID times and 88% during COVID times, which is not significant changes. And similarly with the change in NIH stroke scale from presentation to discharge were approximately the same. So that was a decrease in about 9 points in the NIH stroke scale, respectively. And the discharge location did not reach statistical significance. However, there are definite trends in patients going more to home than to acute rehab during the COVID era. And that makes sense because a lot of the acute rehabs were not accepting these patients post-procedurally. Now, looking at the COVID era subgroup themselves between COVID positive patients and COVID negative patients, looking at the individual time points, they were not significantly different. However, when you're looking at the full time frame from last and well to ultimate reperfusion time, patients who were COVID positive had a significantly longer period of time until ultimate reperfusion. And I think a majority of that comes about with the last and well to neurointerventional consultation time, which is about 130 minutes more than the COVID negative patients. Interestingly, these patients who were COVID positive did not, most of the positive results came about afterwards. So just an interesting note to make. So then in terms of discussion, in summary, patients during the COVID era were younger, overall healthier with less atrial fibrillation, and then a better functional baseline. We found that despite strict social distancing, overall the workflow times between pre-COVID and COVID times were not significantly different. And we had essentially equivalent functional outcomes and radiographic outcomes. And then lastly, the COVID positive patients do have a longer time to presentation, which is not significantly different statistically. However, there was a trend towards that. But there was an overall longer time flow from last and well to ultimate reperfusion in COVID positive patients. Thank you very much for your time. I think there's a lot of interesting results here, and we're going to be definitely looking more into this and trying to pick out why exactly this may have taken place for these patients. Thank you very much.
Hey, Kurt, this is Jay. Why do you think the COVID patient population, I know you just said you're going to look into it more, but why do you think they had these differences? Why did you see patients that were younger? Why did they have higher functional status to start, although that follows with them being younger? Why did maybe more of them go home? Do you have any thoughts on that? By the way, this is awesome data. Bits and pieces of this I've seen before, but I haven't seen it all. As a really fantastic synthesis of everything.
Yeah. In terms of the functional outcome, I truly don't know. I mean, we saw a lot of young people as Tom was able to present to the New England Journal. We saw young people. We saw a lot of different boroughs being represented in our cohort. People from different institutions we had never seen before. I think during the times everything was a little bit chaotic, and so we were fortunately able to bring in a lot of people to our institution to help out. In terms of why the younger people, I think we're going to have to look at those patients specifically and look at their lab values and look at their maybe some other underlying comorbidities, but I truly don't know about the age.
Well, let's say it differently. Do you think that COVID itself is causing people to have strokes, which is why we're seeing it in younger people, because we see younger people with COVID now, or we were seeing younger people with COVID?
Yeah, that I'm not sure. I don't know.
Hey, Kurt, I was going to add to Jay's comment. I know Jefferson and Philly had a surge of these types of cases. Were you able to kind of reach out to other centers across the country, like in Detroit and other places where there are COVID hotspots, to confirm this too?
Yeah, that's the next step. We're going to look into, first starting in New York City, the different institutions to really get a bigger population. As I said, NYU published a series suggesting that the stroke rates were actually lower during the COVID time. They had some methodological flaws, but looking past that, they also confirmed the lower age of stroke patients during this period. But I think once we have a greater population, because our cohort was, there was a larger number of LVOs, large vessel occlusions during COVID. So the two institutions really are not finding consistent results in that regard. So I think compiling these numbers to get a better answer, and I think bringing in institutions from other areas and different hotspots would also be really important.
Awesome. Nice job, Kurt.
Thanks a lot, Kurt. Let's move on to Robbie Rothrock. Kurt, can you stop?
Yeah.
Where do I click on this one?
Okay, Robbie, we've got you there.
Hey, good morning, everyone. Can you hear me?
Yes.
Great. I'm Robert Rothrock. I'm a PGY6 here at Sinai. I'm going to change gears from vascular and talk this morning about our experience with topical TXA for spine tumor, extradural spinal tumors, which basically over the last 3 years we've been doing with Dr. Caridi and Dr. Margulis, Dr. Zoe. So I'll speak briefly about the background on TXA and topical TXA, the surgical technique that we've used, and then finally discuss the results that we've had here at Sinai. I have no personal disclosures. TXA has basically every day, almost it seems, an expanding number of clinical indications. It was originally FDA approved for uterine bleeding in OB/GYN. However, it's been long used in orthopedic surgery, trauma surgery, and even spine, and usually in a systemic administration, intravenous administration. But because there's great safety data for TXA, it's widely used off-label for a number of other indications. And more recently, it's been utilized topically, especially in the orthopedic joint literature, as well as other forms of orthopedic surgery and OB/GYN surgery. And one of the nice things about topical use of TXA is that it can help mitigate rates of DVT and venous thromboembolism in specific surgical populations because the apparent dose systemically is much lower when used topically. Along these lines, it lends itself very well to the risk profile of metastatic and primary spinal disease. We know from a large study out of Sloan that the preoperative incidence of DVT in all comers for metastatic spinal disease is almost 10%. And in patients who have compromised ambulation, that goes to 25%, just about. So this is an at-risk population for VTE events. So the other complicating feature is that many centers do not like to use technologies such as cell saver, so-called blood salvage systems, because there is a risk that as you suction and then re-administer those products, you're delivering cancer cells systemically. There are filtration systems, but they're not available at every center. In fact, I don't think we have them here at Sinai, although we do have them in Elmhurst. So the thought is that if we can use topical agents such as TXA, in addition to Surgiflow, other things that we use, we can help mitigate these relatively high blood loss surgeries. So the technique that we use here is we take 1 gram of TXA, which is found on virtually every anesthesia cart in the hospital, is relatively cheap medication, and we dilute it in 1 liter of sterile saline or plain irrigation. And then, as you can see in panel D, we'll take Ratex or gauze, soak it in this solution, and then use this to pack the gutters, as you can see in panel E, as we do the other portions of the surgery in other areas. So we looked at all the spine tumor patients that we've used this on from October 2017 to November 2019. And in this period, we were using this fairly regularly. We included patients with solid tumor, metastatic disease, hematologic malignancies such as multiple myeloma, or primary malignant extradural spine tumors, so sarcomas, primary tumors that were not intradural. Every case that was included involved open posterior decompression with tumor resection and instrumented fusion, so either separation surgery or corpectomy. And we excluded patients who were having a minimally invasive approach, pregnant patients, which thankfully did not exist in this cohort, and any patient that had a coagulopathy that would preclude using a procoagulant agent. Primary outcome we used was 30-day risk of DVT and pulmonary embolism, but we also looked at a need for transfusion, postoperative blood loss, intraoperative fluids needed, postoperative seizures, because TXA, if in contact with the CNS, can lower the seizure threshold, and any other non-routine postoperative complication, length of stay, and 30-day mortality. So most of our findings were in line with what's been reported in terms of demographics. On average, patients were more male than female. Average age was around 60. There were no patients that had preoperative history of seizure. There were some patients that had liver disease, renal disease. Overall, ASA status was elevated because these are patients with cancer, so most were 3 or above. In terms of the spread of malignancies, we had a variety from breast to myeloma to chondrosarc and giant cell, so a fairly large range of pathologies. Most of the surgeries that were performed were large surgeries, so the median number of segments fused was 5.5, with usually at 3 levels getting laminectomy or decompression. 5 of the cases involved corpectomy, 11 were simple separation surgery. You can see that the mean blood loss was over 1 liter, just over 1 liter, and 4 patients had a delayed extubation, meaning they had to be extubated outside of the operating room, so in the ICU usually, or the PACU. In terms of the drains, the mean total drain output was also over 1 liter, so these are patients with number of indwelling hemovacs with substantial postoperative output as well. In terms of the primary outcomes, we had 1 patient with a DVT within 30 days and 1 patient with a pulmonary embolism within 30 days, no wound infections within 30 days, 2 patients that had durotomy and CSF leak, 0 requiring reoperation, no 30-day all-cause mortality, and a median survival of 6 months, which is consistent with this surgical population. We also had a median follow-up time of 6 months for survivors. This is just showing. Say that again, Chris?
Less than a minute.
So just to conclude, in our cohort, we were fairly in line with the reported rates of DVT and PE. 2 studies, larger studies included here, had 17% and 4% DVT rate and a 3 and 1% rate of PE. Also, in terms of blood loss, we actually, although this is a large amount of blood loss, we were less than the reported rates in literature overall. So although this is a small retrospective study with a number of confounders, really the goal was to show that this is a safe and feasible adjuvant to intraoperative hemostasis. Obviously, a randomized controlled trial will be needed to better assess this, but in lieu of that, we can continue using it off-label, we think. I'll take any questions that audience may have. I guess it was very convincing.
Rob, this is Jay. I think that was great. Do you think, I mean, what are your next steps? Where do you want to take this research aside from just doing it off label? What do you think would be the right next way to go?
I would love to have either a small scale randomized feasibility trial or just to start a clinical trial. I think it's great safety data. There's enough safety data to do a clinical trial, and it doesn't seem to increase risk. I think this could be used as preliminary data to move forward with the trial, and I think we could have a multi-site trial pretty easily. Yeah, I think everyone tries to mitigate this type of blood loss during these surgeries, and anything that's safe is welcome. So that was what I would like to do and carry it forward.
Well, the next step for you would probably be to pursue an IND to do a trial. You've got 2 of your faculty in this department that have held or do hold INDs for this kind of work. You should engage and see about setting that up. It's a pretty straightforward thing when you're doing from from a physician side and from a clinical indication side.
Yeah, I'd love to do that.
I don't know, John, are you on? Do you have any comments or questions?
So, this isn't necessarily a new thing. It's just that it was new in tumor surgery. So I do agree that, our next step would be to do an IND and then start a clinical trial from there and, work toward, making this at least something else that people have in their armamentarium for this kind of surgery.
Very cool.
All right, thank you, Robbie. And Jeff Gilligan. Robbie, can you stop screen sharing?
Yes, I can.
All right. Can you see that okay?
Yes.
All right, great. Good morning. I'm Jeff Gilligan. I'm BPGY5, and I've spent this last year with Dr. Creedy as a fellow. Today, I'll be presenting on hospital frailty risk score as it predicts outcomes for cervical fusions. I have no personal disclosures. So some background. So frailty is a utility that we use to define basically a multidimensional syndrome of loss of reserves, such as energy, cognition, health, that gives rise to vulnerability and potentially adverse outcomes. In surgical populations, been more utilized recently as a way to help stratify risks in particularly elderly patients. And currently, there's been several different frailty scales that have been developed as a way to predict and identify patients at increased risk of adverse events, but none has emerged as a gold standard. And here's just an example of one called the clinical frailty scale. The advantages of one like this, it can be used by any clinician after any encounter, such as clinic, and they can rate it on a 1 to 9 scale. And it's a way to basically screen for certain patients who are going to be at risk for more adverse events. The downside of this is it's a subjective method, and it adds an extra step after seeing a patient. It's not an automatic thing. So the hospital frailty risk score was developed in 2018, and it's based on a novel method of assessing frailty utilizing ICD-10 codes. The advantage of this score is that it can be implemented just using the hospital information systems, removing the interoperator bias or variability, and provides a low-cost systematic way to screen frailty. The also advantage is that it can improve efficiency to analyze large data sets for frailty and automatically update the score throughout a patient's life. And the way they created the score was they identified a cohort of patients who already had markers for frailty, such as dementia, falls, and they found associated ICD codes and developed this list of over 70 ICD codes that can be utilized in the score. And what they found is that this frailty score was associated with increased mortality, length of stay, and readmission. And they divided the score based on 3 different cohorts as low risk, intermediate risk, and high risk. So our objective was to use this new scoring system for frailty and assess how it would be able to predict complications and outcomes in cervical spine fusion surgeries. So we utilized the national inpatient sample from 2013 to 16 and looked for procedural codes for ACDFs and posterior cervical decompressions and fusions. You can see we had more ACDF patients than posterior decompressions. And then using the same way, we divided the cohorts into low, medium, and high frailty groups. We then assessed 15 common postoperative complications, as well as the length of stay and total costs, of course, across these cohorts. And we assessed the predictive power of this method by adding it to a base model, which looked at age, sex, primary pair, race, and comorbidity burden, and then assessed it using a under the curve operating characteristic. So here are the cohort characteristics. In the ACDF group, the low frailty score were more likely to be younger, female, have less comorbidities, private payers, and non-white, which is kind of what we already kind of would assume. Same as posterior cervical decompression. And so then we assessed the difference in the cohorts for complications and the length of stay and cost. And as you can see, as the frailty score increases, the length of stay, cost, mortality, as well as all these different complications increases dramatically. And interestingly, between even the low and medium and the low and high, there's a stratification. So there's the advantage of this score is that it even has the nuance of as it slowly increases, the different complication rates increase as well. We had a similar finding in our posterior cervical group as well, where the length of stay, cost, mortality, as well as the complications increased between low and medium and the low and high groups as well significantly. So then we looked at the predictive ability of the scale, comparing it to that base model, which used the age, race, payer, and comorbidities. And the model with the hospital frailty risk score included had a much better predictive methodology compared to just the base model. For the ACDF, it was better at predicting 10 out of 14 postoperative complications, including mortality. And in the posterior cervical group, a little bit less so, it was 8 out of 14, but still shows its utility as an improved way to predict these postoperative complications. The limitations of the study, it's retrospective. The NAS data set also doesn't include important variables such as specific disease pathology or baseline neurological deficit. The scale does have its downsides. It can't capture complex patient characteristics as dynamic functional states or whether or not someone's being taken care of by another caregiver or fluctuations influenced by acute disease. It also doesn't include ICD codes for diabetes, cardiovascular, pulmonary disease, which are already known to be risk factors in spine surgery, especially in the elderly. But in summary, the hospital frailty risk score was a positive predictor for length of stay, mortality, adverse events, and total hospital costs for both anterior and posterior cervical spine surgery. And going on this, it could has the potential to be a value predictor in length of stay costs across other populations in spine surgery. Just want to acknowledge everyone, Dr. Creedy, all the help in his group, Sean, Rebecca, Eric, Dr. Gall, and everyone involved. Thank you.
So frailty has definitely been a very hot topic amongst all of our specialties, all surgical subspecialties in assessing our patients and their outcomes. And I think this scale is very nice in that it allows us to use basically data that's already been collected and put it into an algorithm to come up with a very usable frailty scale to predict the outcomes of these patients. And I think the long-term goal is when we do move to a more episode-based cost model with the payers, that this will be something that could be factored into that to help increase reimbursement or help us out in that way.
Okay, Jeff, can you stop sharing your screen? And Jacques?
I'm here. You just. Do you see my screen?
We see you.
You see my screen now?
Yes.
Okay, good morning. My name is Jack Lara. I'm one of pre-residency nursery fellow at Mount Sinai Hospital. I'm presenting staying home early changes in patterns of neurotrauma in New York City during the COVID-19 pandemic. As we know, COVID-19 has shut down the world. By March 11, the World Health Organization declared this disease as a pandemic. In February, United States found their first cases, and New York City has been the most affected city around the world, with an overwhelming number of patients of 6,000 cases, new cases diagnosed in a single day. In an attempt to decrease the rate of infection and progression of the COVID, the New York State, city, and state governments implemented social distancing and restriction of non-essential activities starting on March 12th. Our hypothesis was that once these policies are implemented, we expected to see a decrease in numbers of neurotraumas among the city. This was an observational, retrospective, and prospective analysis of all the neurotrauma admissions and consults from November 2019, the moment that the pandemic started in China, to April 2020 at Morningside, which is our designated trauma center. We dichotomized this data in pre-COVID era from November to February, and COVID period from March to April. The variables that we use are general demographics, mechanism of trauma, radiological diagnosis, and treatment instituted. We perform a comparative analysis between these two cohorts. Overall, we found 150 patients were seen in our service from the total length of the study with neurotraumas, and a mean age of 66.2 years and a male prevalence of 66%. The most common mechanism were mechanical fall in 54%, unknown mechanism in 15%, violence-related or inflicted trauma in 10% of the cases. The most common diagnoses were subdural, traumatic cerebral hemorrhage, and intracerebral contusions in 48%, 26%, and 10%. And the most common intervention that these patients required was medically management only in 82% of the cases. Once we dichotomize the data, the most relevant findings is that more males sustaining a neurotrauma in the COVID period of time compared with the pre-COVID period of time. 77 in the COVID compared with the 60% in the pre-COVID. Also, we found that mechanical fall was the most frequent mechanism in both periods of time. This experienced a drop in the COVID era. Also, we found that an interesting point is that motor vehicle accidents and direct trauma with an object of violence related increase in frequency in the COVID period of time, from 5% for motor vehicle accidents to 18%, and from 9.9 to 12%. All these differences reach statistical significance. Diagnosis, as I mentioned before, this did not change and didn't reach a significant difference. And medical management, it was the most common intervention in both periods of time. This graph, we can see the monthly frequency of neurotraumas from November to April. And as we see here, April has the lowest number of cases. In this graph, I put together in a timeline from the first time, the initial outbreak in the city in the first week of March. I put the COVID patients among the city. As we can see, experience an exponential growth of the cases. And also I put all the neurotrauma cases in the same period of time. By the second week of March, the World Health Organization declared this as a pandemic. The third week of March, the government of New York State and city implemented these social distancing policies. And as we can see here, it wasn't at this moment that the frequency of neurotrauma experienced a drop and continue until the first week of April. In this week, it was specifically in April 6th, it was the highest number of cases diagnosed in a single day in the city. As we know, as we experienced that mechanical fall was the most common mechanism of trauma in both cases. These in both periods, this decrease in the COVID era. As I stated before, the motor vehicle accidents and transit-related accidents increase in the COVID era, as well as violence-related. The literature that I review mentioned that this is not isolated only to New York City. Some other cities around the country has been noticed an increase in motor vehicle accidents, and this may be explained as less traffic in the roads, and people who are actually driving, these people tends to drive faster if there's no traffic around. Also, related to violence, some reports said that the fact that the persons are in isolation, there is increased risk of vulnerability to suffer domestic violence, specifically with these isolation requirements. Something that is important to know but did not reach a statistical significant difference is that more people during the COVID period was converted to a DNI/DNR status from 5.9% in the pre-COVID to 12.2% in the COVID. Unfortunately, this did not reach significance. And as I mentioned in the graph, we noticed an inverse relationship between the number of positive cases diagnosed during this crisis and the frequency of neurotraumas since March 1st. As a conclusion, we noticed that there was a temporary and progressive decrease of neurotraumas during this the initial phase and the peak of the COVID crisis in the city. The tendency to decrease in the number of trauma was observed in conjunction with the implementation of social distancing policies as the number of total infected cases increased in the city. Also, we said that more males sustained neurotraumas in the COVID-19 period. Mechanical fall was the most common mechanism in both periods, and motor vehicle accidents and violence related were seen more frequently in the COVID period. Thank you. Any question?
Jack, we found that there was an increase in violence-related trauma. Do you have any comments on that? Why could this happen during this period?
As I review in the literature, and it's something that could be expected. There's in isolation also the stress that the people experience during this crisis, not only related to the fact that you are in an isolated environment, but also the fact that people are losing jobs, still have debts. All these kind of stress factors may lead or may predispose to an aggressor to be more tend to commit violence towards the people who he is in isolation with. That's what I read in the literature.
Jack, do you think that fear of coming into the hospital played a role in the decrease at all? Or do you think that if someone has a neurotrauma, of course they have to come in?
That's also one of the limitations that we found, as we it's not only related to neurotrauma, because we noticed that also in stroke, and the people who are not experiencing too severe symptoms, this person may not wanted to go to the hospital. For example, mild traumas that are not reported in this cohort, because people are not going to the hospital by fear of getting the infection in hospital, even with a sustaining a trauma. So I think that people was afraid to go to the hospital even with sustaining any kind of neurotrauma.
It's really interesting. Be interested to see how that plays out if there's a second wave.
Yeah. I think now with with the protests. I think that's a high risk also to start like a second wave, maybe. Who knows? Hopefully not.
Okay, our next speaker is, let's see if do we have a question in the Q&A box? Yes, we do. Devaldo Camera, why do you think there's an increase in trauma cases in mid-April since people were still social distancing?
That's a good question. It's hard to say because, first of all, it's not a mandatory restriction. It's people can still go out. The thing that are restricted specifically are the non-essential activities, like restaurants are closed, entertainment kind of activities are closed, but people still, you can go, even not now, but in mid-April, you can see people hanging around in the parks, walking around, biking. So this is something that you can see, and that this may explain a slight increase in the neurotrauma cases in the mid-April, even with the social distancing policy that they're still in place. This is not like a complete a mandatory restriction. It's pretty much like a recommendations of social distancing.
Okay, thanks, Shaf.
Thank you.
Michael Martini.
All right, can everyone hear me all right?
Yes.
Okay, and share screen. Can everyone see the screen all right? Okay, so good morning. My name is Michael Martini. I'm an MD PhD student here at Mount Sinai, and today I'm excited to present on some of the work I've been doing with Dr. Creedy's group, focusing on developing explainable machine learning models in spine surgery. This particular project uses principles from cooperative game theory to interpret models of non-home discharge after spine surgery. So just some quick background. Following spine surgery, many patients experience a long, difficult recovery process, which can sometimes require discharge to specialized facilities for rehabilitation. However, many studies in the spine surgery literature have now found an association between non-home discharge and certain adverse post-operative events like complications and unplanned readmissions. For this reason, predicting which patients are more likely to benefit from additional attention and resources post-operatively is essential to spine surgery optimization. To this end, previous studies have examined individual factors associated with NHD. However, no study has used explainable AI models to do this, nor have they focused on interaction effects between these factors, as they may modulate overall NHD risk. So in this study, individual machine learning models were created to predict non-home discharge for each patient in our study population, which was over 11,000 patients, using over 90 variables collected from their hospitalization records at Mount Sinai, which was for a degenerative spine condition. Shapely values were then calculated for each model to identify important model features and interaction effects that underlie the NHD risk. And just briefly, Shapely values are widely used in game theory to understand which contributions each part of a model makes to the total model output, making it very effective for machine learning interpretation. Now, I won't go too much into the complicated mathematics behind their calculation, but instead, I think one way to conceptualize them is as follows. So imagine any type of team-based game that you want. It'll likely involve players, a set of rules that in turn result in consequences and payoffs in the game. Now, abstractly speaking, each player in the game might have certain skills or abilities that confer increased or decreased value to the overall outcome of the team, such as winning or losing. In addition to their own value in the game, you can imagine that certain combinations of players on a team might work better or worse than others, thereby impacting the team's final result. So in this way, we can apply these principles to medicine, whereby a patient's hospitalization is considered, quote unquote, the game. The patient's characteristics are the players in that game. The rules are the protocols and procedures in the hospital. The consequences are adverse outcomes, and payoffs are good outcomes for the patient. So after calculating the Shapely values for each patient in the study population, we rank them by their overall impact on the model output, shown as the x-axis there on the left. So here, each dot represents a patient in the population, and the color of the dot represents the value of the feature in question. So this analysis showed that higher values of length of stay, age, Medicare insurance, ASA, and estimated blood loss, among others, were among the most important features for higher predicted risk of NHD. So how do we know if we can trust these numbers though? So that's where on the right, you can see the ROC curves for these models. And the important curves in this plot are the purple and yellow ones, where the purple one is the predictive capacity of the model when it's able to access all 90 variables that were in the data set. The yellow one, conversely, uses the top 15 most important variables that were identified there on the left from our Shapley analysis. And the high and similar values of the area under these curves, 0.9 and 0.91, suggest that these models are accurate and that the Shapley values are accurately identifying the most important model features that are driving accurate non-home discharge prediction. So with these Shapley values in hand, we saw an opportunity to conduct a more global analysis of our study population using these values to perform supervised clustering. On the top, you can see what is termed a force plot, representing what goes into the predicted non-home discharge risk of a single patient. So in red are characteristics that are pushing the predicted output higher for this one patient. So in this patient, having Medicaid and a high ASA are driving their predicted NHD risk higher. In blue are the characteristics driving the risk down in the patient, such as their shorter length of stay and their non-elderly status. Now, if you take that force plot, rotate it by 90 degrees vertically, and repeat that for all 11,000 plus patients in the study population, and cluster them by similarity, you arrive at the bottom figure, which depicts the predicted NHD risk across the entire study population. Now, interestingly, with this analysis, we can clearly identify several patient populations of interest, including those who have known risk factors for non-home discharge, yet display an unusually low predicted risk of it. One example is a cluster of elderly patients who have a low NHD risk. And as it turns out, that cluster also shared low estimated blood loss and a short length of stay, suggesting these other features may be interacting to modulate this cohort's overall NHD risk. So this brings us to our interaction effects analyses. While we calculated Shapley interaction values for every set of variables in our data set, in the interest of time, I'll only show the results from a more focused analysis here, which we thought was more clinically relevant. So in the spine literature, both age and comorbidity burden have been recognized as risk factors for adverse outcomes after surgery, including non-home discharge. However, while they're often concurrent in patients, the literature is conflicting regarding which of these represents the more serious risk factor. And to provide a new angle to this discussion, we compared the interaction effects driving NHD risk between age, ASA, which we use as a proxy for comorbidity burden, and 3 different measures of surgical invasiveness, including the number of spinal segments operated, estimated blood loss, and the length of surgery. And our analysis suggested that age was the comparatively bigger risk factor because its interaction effects, which are shown here as vertical dispersion, were around 5 to 10 times the magnitude of those interactions for ASA status. So just to conclude, Shapley values represent a powerful method for identifying important features for accurately predicting outcomes after surgery and for measuring interaction effects across a large study population. Our focused interaction effect analysis suggested that meticulous intraoperative hemostasis may be particularly important for reducing non-home discharge risk in the elderly. And at the same time, patients with multiple positively interacting risk factors may be deserving of special attention and monitoring to mitigate their risk of adverse outcomes after surgery. So finally, I just want to thank the members of our research team who made this work possible. I especially want to thank Dr. Caridi for his fantastic mentorship and guidance. I also want to thank our department leadership for their support and for helping to cultivate a scholarly environment here. Thank you for your attention.
Michael, that's great. This is Jay. What about, did you look at the interaction of the variables, or does this modeling, or is it able to pick that up? Could the blood loss be actually an indirect indicator for the multiple, aspects of surgery or other things? It comes out as the relevant one in your model to be signaled, but it could be due to interaction or sort of a associated variable with some other factor. Have you thought about that or looked into those things?
So, I mean, that's kind of why we looked at other measures as well. We looked at the duration of the surgery and how many spine segments were operated. So, I mean, these are all kind of, in our eyes, looking at somewhat similar things, basically how intense of a surgery did the patient undergo. And we saw the effect pretty much throughout all of these. And so it's not necessarily just estimated blood loss, but that one definitely did show some important and significantly large effects. I definitely think that this is something that's deserving of a more focused analysis. So one thing that is on the conclusion slide that I didn't mention in the interest of time was that there are certain limitations with the data that we used. So we used a large data set derived from Mount Sinai's data warehouse. And unfortunately, we lack a bit of granularity in some of that. So that's definitely something that I would like to follow up with on maybe like a smaller scale, looking specifically at patients and variables of interest and really trying to deduce what these actual effects are. But yeah, so age and estimated blood loss did have a very strong positive interaction effect, as you can see here with the increased blood loss and increased age having the highest contribution towards non-home discharge. But at the same time, despite having a higher age, low estimated blood loss actually dramatically decreased the risk factor that age posed on non-home discharge risk. So that was kind of why we made that suggestion in the conclusions. But that's a great question.
Mike, this is an awesome study. I think it shows that, you know, we can't just think of, you know, one magic variable that, you know, is going to predict how our patients are going to do, and that it really is a continuum of looking at all of the variables and how they interact and weighting them differently. And even though intuitively in all of our minds it makes sense, I think that this was a phenomenal way to put it all together and show it mathematically. And I love this work, and I'm glad that you're continuing it.
Thank you. Yeah, I think one way to possibly continue the work is to eventually develop more of a predictive algorithm that can be possibly deployed in a clinical setting so that we can maybe triage patients accordingly, give them any extra attention that they need if they do have multiple positive risk factors. As you were saying, it's not just only looking at elderly patients, it's looking at elderly patients in combination with one thing or the other, any other subset of patients. But
hey, Mike, Kostas Sajapanias here. How about looking at with comparison to the frailty index? Is that something worthwhile to analyze and validate some of your studies?
Yeah, absolutely. I think so that I don't think we had done those calculations and calculated like a frailty rating for each patient when this database was built. But definitely now that we've done that calculation in the group and we can assign a frailty risk score for each patient in our cohort, I definitely think that would be something to look at, see what interacts with frailty. We might see a lot similar variables as age and ASA, but at the same time, we also might see some new things that you know could be more useful clinically.
Nice job.
Nice job, Mike. This is Dolores. I have a quick question. Very nice talk. So I have a question. You said you reviewed 11,150 cases. So is it database was collected for some other reasons and you just use it historical data set? I'm just trying to see if there was a bias in collection of the data set.
So yes, so I believe this is actually from Mount Sinai administrative data.
Yeah, I can. It's data collected from anesthesia.
Oh, okay.
And it is a lot of administrative data combined with what anesthesia generally collects and warehouses for each patient. So it's from not only Epic but also the anesthesia records. So this is not necessarily data that we're you know, I guess cherry picking in a way. It's data that's independently collected.
Oh, great.
Yeah.
Thank you.
And I should have mentioned this, but the only cases that we really excluded were those for traumas, tumors, and infections. So we tried to isolate as closely as possible patients that had a degenerative spine condition.
Great, thank you.
Thanks a lot, Michael. Let's move on to Christina. Michael, can you stop sharing your screen?
Yes.
Okay, I'm gonna share my screen now. Okay, can you see that okay?
Yes.
All right, hi everybody. My name is Christina Rosito. I'm a first year medical student and I'm here to share our findings on the time delay from image acquisition to INR notification within the acute stroke workflow. Stroke thrombectomy has become an accepted standard of care for acute ischemic stroke caused by emergent large vessel occlusion. Its efficacy relies on expedient treatment times. For example, the SWIFT Prime trial showed that functional independence decreased by 10% over the first hour of treatment delay and by 20% with every subsequent hour. Public campaigns like Be Fast promote expediency of stroke identification. Diagnostic accuracy by the general public and EMS has been improved by stroke severity scales. Mobile stroke units with portable CTs have begun to enable accurate field diagnosis and improve triage. Hospital transfer bypass protocols have begun to been implemented in several regions of the country, and pre-hospital notification of patients with suspected stroke has also been found to reduce time to treatment. However, there has not yet been a focus on the window from image acquisition to INR notification. Therefore, we hypothesized that the time from radiographic LVO confirmation to neuroendovascular team activation makes up a significant yet modifiable portion of time spent within the acute stroke workflow. For those less familiar with the stroke workflow, EMS patients arrive at the hospital and are first taken to CT. From CT confirmation, the INR team is notified, and then the patient goes to the angio suite. There, thrombectomy occurs from groin puncture to reperfusion. For transfer patients, additional time is spent on the transfer from the peripheral hospital to the central hospital, and the INR team is typically notified before imaging at the central hospital. Here, we looked at Sinai patients with LVOs from 2017 to 2020. Due to the altered workflow shown on the previous slide, we excluded 322 patients who were transfers and inpatients. 12 patients were excluded due to missing timestamps, and 36 patients were placed into a pre-notification group because the neuroendovascular team was notified before CT acquisition, usually based upon clinical presentation. The remaining 143 patients were then dichotomized into rapid within 20 minutes and delayed over 20 minutes notification times. This time point was based on the American Heart and Stroke Association's best practice guidelines, suggesting a goal of 20 minutes from initial CT scan to neuroradiographic interpretation. Looking at the patients from the rapid, delayed, and pre-notification group, here's an overview of the patient characteristics. You'll see that the mean time from CT to INR was 29.4 minutes, near the guidelines of 20 minutes, and making up 23.8% of the total time from presentation to reperfusion, a significant and targetable window. Now looking at the bisected data based on the American Heart Association guidelines for CT imaging to INR, you see that the average time window for the rapid group was only 8.7 minutes, and for the delayed group, it was 81.6 minutes. We found that the mean overall times from hospital presentation to reperfusion were 155 minutes and 269 minutes respectively, leading to a time difference of 114 minutes between the rapid and delayed groups. So the proportional time delay caused by lagging imaging notification increased from 6% in the rapid group to 30% of the total time delay until reperfusion in the delayed group. Interestingly, if you look at the top, this is the pre-notification group, and it yielded a final reperfusion time of 138 minutes and could represent a subgroup of implementing early communication policies. The time differences between the rapid and delayed groups may be explained in part by the condition of patients presenting. Patients in the rapid group had a better functional baseline with pre-stroke MRS 0 or 1 at 74.6%, compared to only 60.2% in the delayed cohort. This trended towards significance and could be interpreted as easier stroke recognition in better baseline patients. The rapid group had significantly higher presenting NIH stroke scores. Therefore, it's possible that more severe cases were easier to recognize and expedited, while cases with lower unclear severity were delayed for further imaging interpretation. Other sources of delay were likely due to inefficient communication and systemic barriers. Given our results, solutions are needed to expedite the notification. One potential solution is pre-hospital notification based on clinical suspicion for LVO. While this may lead to faster treatment, the rate of false notification results in wasted resources and team burnout. And relying on this will still miss patients with mild symptoms who are at risk for worsening. Process improvements include identifying at-risk time periods, enhancing team communication, and faster recognition of abnormalities within imaging. Software solutions have emerged to deal with image identification and communication. Machine learning algorithms quantify stroke core, penumbra, mismatch, branch occlusion, and predict complications. Current software platforms include Viz.ai, Brainomix, and more. Viz.ai received de novo regulatory clearance from the FDA in 2018 for the first computer-aided triage and notification platform to identify LVO in CT imaging. In conclusion, time delays from initial CT acquisition to neuroendovascular team notification can prevent expedient treatment with endovascular thrombectomy. Process improvements and automated stroke detection on imaging may ultimately improve time to reperfusion. Thank you.
Christina, this is Jay. Excellent position. I don't know if Chris has any thoughts, but where do you see sort of the technology? You sort of highlight that faster recognition with technologies may be helpful. Do you think that there are more, what about, these are all things about notifying physicians, sort of still working with a normal plan. What about getting the treatment to the patient in a better way? Robotics, these kinds of things. Do you think that's going to have a role in shortening this critical time period?
I think that could play a role, but I think that ultimately, in my opinion, it's important to have a physician there, and so I think that improving the notification to the physician is the most important way to reduce this delay.
Well, actually, I got to agree with you on that. I think that's a good point.
Yeah, I think, thanks, Christina. This is Joanna. I guess the question is, how do you think there's the importance of the accuracy? You know, how you mentioned that false notifications. How do you think this kind of weighs in on the team? Like, how accurate does the prediction of LVO have to be before we can start using it to notify teams? Like, if we go with the clinical score, is that enough, or do we have to wait for imaging?
Yeah, no, I think that's a really good question, and I don't have an exact answer. I think there's a lot of different components to weigh. I definitely think that whether it is the severity scales which are being used or future machine learning programs. Those, they have to be tested and really verified before they're used. And I think that as they're used in clinical trials more and more, that accuracy will improve.
Yeah, I agree. I think in the end, it has to do with if we said we were going to notify everybody based on clinical suspicion, we talk a lot about well-being and burnout, and I think that's a large part of the reason why it's not very practical to pre-notify the INR team on every suspected LVO.
Mm-hmm. Yeah, I definitely agree.
Thank you very much, Christina. Can you stop sharing your screen? And George, are you on? Should be at the top.
Here we go.
Hi, I'm George Maragos. I'm one of the pre-resident fellows. I hope you can see my presentation here. Thank you very much for the opportunity to present to you today. I'm going to present my research with Dr. Hambardzumyan about fluorescence-guided high-grade glioma surgery using 5-ALA, and specifically for the timing of 5-ALA being more than 4 hours before induction to anesthesia.
Hey, George, you're in presentation mode, so we can see the notes in the next slide.
Let me change that. There we go. All right.
Great.
So these are our disclosures. And let me just start by talking about high-grade gliomas and the reasoning behind 5-ALA for these in the first place. So high-grade gliomas, the extent of resection for these tumors can directly impact patient outcomes and overall survival. However, these tumors are very heterogeneous. They have very invasive behavior, and they look very similar to the surrounding parenchyma. So they're very tough to actually take out completely. So this is where 5-ALA, 5-aminolevulinic acid, comes in. This is a substance that the patient drinks, and then it's metabolized to fluorescent protoporphyrin 9, which is taken up preferentially by the malignant glioma cells inside the brain. And we can intraoperatively these fluoresce, and we can differentiate where the tumor is and where there is normal tissue. So this, the 5-ALA got FDA approval in 2017, which is actually 10 years after it got approved and was used routinely in Europe. And the basis for this, for the FDA approval was the randomized controlled trial by Tumor and colleagues, which involved 322 patients randomized between 5-ALA and conventional microscopic surgery. And there was benefit for 5-ALA, both for complete resection and 6-month progression-free survival. Now, currently, the FDA administration guidelines dictate that 5-ALA has to be given between 2 to 4 hours prior to induction to anesthesia. And the basis for this is that the previous randomized controlled trial that I talked about used exactly this timing for administration of Gliolam. And the more scientific basis is that 5-ALA has its peak concentration in the blood at around 4 hours. However, it does take time to be metabolized to its active fluorescent component, protoporphyrin 9, and there is no clear evidence about how long it actually stays inside the glioma cells and how long it actually fluoresces intraoperatively. So for this study, we wanted to demonstrate that there is adequate intraoperative fluorescence in cases where surgery, where induction to anesthesia took place more than 4 hours after 5-ALA is administered to the patient, and as is FDA approved. And of course, our secondary objectives were to assess the clinical survival and the radiological recurrence rates for these patients, and show that they are not inferior to standard cases where 5-ALA is given less than 4 hours before induction. So for our methods, this was a retrospective case series between the years of 2019 and 2020 in Mount Sinai Hospital. We included patients that were induced to anesthesia more than 4 hours after 5-ALA was imbibed by the patient. And we used Oliosphere software to get volumetric measurements of the contrast enhancing part of the tumor in the preoperative and postoperative MRIs, as well as the 6-month MRI to assess extent of resection, as well as 6-month progression-free survival. And our primary outcome was intraoperative fluorescence. Now, here are the baseline characteristics of our patients. I'd just like to draw your attention to the tumor volume. It was the mean tumor volume for these patients was 17.8 cc's, and the mean time to induction to anesthesia was 5 hours and 24 minutes. Now here you can see a video where this patient drank 5-ALA and was induced to anesthesia around 6 hours later. And you can see here that there is very good fluorescence intraoperatively. It was, the borders were shown as normal, and the resection was very good. Here you can see the outcomes of our patients. We had no cases of 5-ALA related toxicity, and residual volume, the mean was 1.15 cc's. Extent of resection was 90.4% as a mean, and 9 of our 13 patients had 6-month progression-free survival, which is 69%, which is comparable to the range of 42 to 80% that is reported about 5-ALA assisted glioma surgery. Of course, there are certain limitations with this study. This is a retrospective study. It's a case series with a small study sample and no control group because we're comparing to the literature. However, it's a first step, essentially, to show that 5-ALA can be administered more than 4 hours prior to surgery. This doesn't show to have a significant detriment to complete resection or patient outcomes. And the main point is, if this is a first step, relaxation of FDA-imposed timing limitations could help with surgery scheduling and or workflow, especially when there are additional cases or category 1s that need to be on the schedule, and we could be more flexible with what FDA has currently imposed. That's my presentation. Thank you very much for your attention. I'd like to extend a special thank you to my mentor, Dr. Hajpanayis, as well as the members of the team, Alex, Rebecca, Panos, Nikita, and Gabriel, for making this possible. Thank you.
Yeah, great job, George. I think the impetus for this study kind of came about from some anecdotal reports of even better fluorescence the longer you wait. And if you look at some of the other fluorophores out there being used globally, this paradigm actually is coming true. And so I think it warrants further study. And we don't know exactly when the longest time period is to wait. Because a lot of the studies are really built on these earlier time frames, but definitely worth looking into. Good job. Thank you.
Thank you.
Hey, George, this is Jay. Do you have a next steps in terms of figuring out what that ideal time period would be?
Well, right now we are also in the process of contacting other centers, Henry Ford, for example, to get their cases as well, to put it together as a bigger case series. We also need to make a comparative study before we essentially need more data to have the power to get significance about when the ideal timing would be for these cases. So we are still forming our initial hypothesis. Dr. Kellner, you're muted.
Thanks a lot. There's a question in the QA box from Nicholas Daddario. How do you believe increasing the time of administration will affect positive or negative predictive values?
I think that is essentially in the same box as what I just said. We don't know what the best time. Essentially, we know when 5-ALA achieves the peak inside the blood, but we don't know what the best timing is for fluorescence inside brain glioma cells. And of course, that's going to also depend on the specific patient, pharmacokinetics, and the glioma itself. So we do need to first form a hypothesis about what could be the best timing, and then we're going to see about positive and negative predictive values for that.
Great, thank you very much, George.
Is there room for another question?
Yes, we do have 1 minute.
Since the extent of resection seems to be related to how good the fluorescence is, and the fluorescence is stronger in more malignant tumors, do you think that the more malignant the tumor, the better the extent of the resection, notwithstanding the more difficulty of doing a resection in a more malignant tumor? How does the grade of the tumor, or the subgrade, the subtype, et cetera, relate to the extent of resection? How are you able to compare the efficacy of 5-ALA without more strictly controlling for that factor?
Thank you, Dr. Patterson. I'm not exactly aware about about the literature, whether there is higher uptake, proven to be higher uptake of the 5-ALA and better fluorescence in higher grade gliomas, which would make sense due to better, to more blood inside the tumor. I'm sure that it's definitely a very good question to put these factors and genomic factors essentially inside our calculations when we're comparing the outcomes for these patients.
Yeah, that's a good response, George. JB, there was a paper in JNS by the Dartmouth group, Dave Roberts, where they looked at heterogeneity of the tumor and gene expression and fluorescence. So there is some story developing there, but for sure, the high-grade gliomas have more protoporphyrin IX production, and hence we can visualize them better with our current technologies. But there are new technologies being developed that can detect fluorescence more sensitively, i.e. use them in low-grade gliomas, which actually produce protoporphyrin IX as well in lower concentration. So there's a paradigm shifts in technology, and there's some gene expression type analyses also being done that, the story is getting more complete.
Great, thank you guys. Let's move on to the next speaker. Thank you very much, George. Devin, great.
Yep. You see that on your screen?
Yes, looks good.
Great. Okay, thanks for that.
Your audio is out a little bit.
Is that better?
Yeah, that's better.
Great. Prior ipsilateral.
Same problem happened again there. Okay, it's good right there.
Okay, how's that?
That's good.
Okay, so today I'm going to be discussing our project entitled Prior Ipsilateral Infarct is an independent predictor of poor outcome after mechanical thrombectomy. We have no disclosures. So the purpose of this study is to investigate the association between radiographically evident chronic infarct and outcomes after mechanical thrombectomy. This entire thing was sparked by the idea that patients who present with chronic infarcts ipsilateral to the side of the treated LVO may be less likely to achieve functional independence afterwards.
Hey, sorry, we lost audio again there.
Goodness. Okay.
All right, that's good.
That's okay. Okay. So again, the hypothesis here was that patients with chronic infarcts ipsilateral to the treated LVO would be less likely to achieve functional independence. By way of background, only modest work has been done in this area in the past, with one study available in the literature that was dedicated to the investigation of the effect of chronic infarcts on good outcomes. Now, it's important to note that this study had no access to data regarding the size of the infarct or location of the infarct, and though they showed a significant univariate effect, the effect on good outcome did not survive after regression for known modifiers. Two other studies in the literature are even smaller with more stringent inclusion criteria and did not select out patients with chronic infarcts from those with acute or subacute prior infarcts. So we set out to design a study that would address some of these limitations. Here I'm showing the first phase of our cohort construction. We had 755 patients identified in the study window, which we set between the end of December 2014 and the end of January 2020. We then applied two exclusion criteria, one of which was that we excluded patients with lack of follow-up at 90 days. I'm happy to talk about this statistical decision in the question section if it comes up. We also excluded basilar and vertebral occlusions, primarily because we're interested on how the side of the chronic infarct affects outcomes. And then with regard to basilar vertebral occlusions, it's unclear which side is going to be affected at the time of pre-procedural imaging. So from these 620 patients, we queried the records and identified 93 with any documented history of prior stroke. This was either per patient report, noted on a radiology read elsewhere, or otherwise made it into the chart. And here we started the second phase of our cohort construction, which was dedicated imaging review by a board certified neuroradiologist, Dr. DeLisi. Here we describe what those imaging inclusion criteria are. We're defining chronic infarct as the presence of a gliotic cavity on pre-procedural CT or MRI, as shown here in the photo to the left with the red arrow indicating a nice example of an MCA dot sign. This incidentally is a patient in whom the prior infarct is ipsilateral to the treated LVO. And conversely here on the right side, we have an example of a patient who is excluded due to the presence of a subacute infarct. We see some signs of cerebral edema, sulcal effacement, and arrows pointing here to what is potentially a small amount of petechial hemorrhage. So having applied those imaging criteria to this second phase of cohort construction, we excluded 10 patients who were found to have evidence of subacute prior infarcts, and 15 of those 93 patients were actually transferred back into the no chronic infarct group. This is because they had no radiographic evidence of stroke. So in the end, we had 68 patients in our final chronic infarct group, 532 in the no chronic infarct group. And again, these 68 patients underwent dedicated imaging review, recording the location of the infarct with regard to the treated LVO, the territory affected, and the size. So here we have a quick look at the demographics and baseline characteristics of our two groups when divided into those with no signs of chronic infarct and those with chronic infarct. Here we have our group sizes, 532 and 68. I won't walk through the entire demographic and clinical characteristics table, but you'll note that we listed characteristics generally known to be associated with the odds of good outcome. And we did univariate testing as shown by the p-values here on the right to look for balancing between our two groups. I'll draw your attention to these three values in red, where we observed that patients presenting with chronic infarcts were more significantly likely to be female, more likely to be older, and nearly twice as likely to present with a poor pre-stroke functional status. This would be important to take into account later.
A little muffled there, a little too muffled.
This would be important to take into account later during multivariate regression. Again, if you hadn't heard me, these three values in red are those values on which the chronic infarct group and the no chronic infarct group were not balanced. So first, we performed a univariate analysis looking at the effect of chronic infarct on thrombectomy outcomes. It's shown in the chart here that any prior stroke did have a significant effect on the likelihood of poor functional outcomes. That is to say, the odds ratio for a good functional outcome was 0.544 with a p-value of 0.04. So here we see again, in our univariate analysis, any prior chronic infarct did put patients at higher risk for a poor outcome. We then did a subgroup analysis where we broke all of our prior infarcts into 3 bins, depending on the location of that chronic infarct. We defined location with regard to the current LVO. So we had 28 patients with an ipsilateral chronic infarct, 23 with a contralateral chronic infarct, and only 17 with bilateral infarcts. And most interestingly, on this analysis, we found that it was only those patients who were presenting with ipsilateral chronic infarcts who were significantly more likely to go on to have a poor functional outcome. Contralateral chronic infarcts had no effect. And interestingly, prior bilateral infarcts only had a trend, though this may be due to the smaller sample size here, only 17 patients with regard to 28 and 23 in the other 2 groups. And important to note that of these 17, only 2 went on to have a good outcome. So this trend may be more due to an issue with power than it is to a real finding. So our final step then, having observed a univariate effect of any prior infarct and prior ipsilateral infarct on thrombectomy outcomes, we went on to perform a multivariate regression controlling for those clinical characteristics upon which our 2 groups were not balanced. And so here we have our final finding where it was shown that any prior infarct was no longer significant after controlling for unbalanced covariates, while prior ipsilateral infarct retained significance.
1 minute, Devin. Oh, great.
And so that leads us to the final conclusion that radiographically evident ipsilateral chronic infarct is an independent predictor of poor outcomes in patients undergoing mechanical thrombectomy. And as far as future directions goes, and we can talk about this more if it comes up during questions, but because we had to break this into a subgroup analysis down to the 24, 28 patients, it would really be nice to have a dedicated investigation of ipsilateral strokes alone, especially if this could be multi-center so that we can improve generalizability, as well as prospective, so we don't have any issues with missing data. We collected here, though we have not yet used in our analysis, data that's much more granular on the territory affected, the size of the infarct, and because of our sample size, we didn't want to start adding in too many covariates for risk of significantly lowering our power. So thank you very much. These are my references at the end, and I'm happy to answer any questions.
I'll just pipe in. It's not really a question. It's just to say nicely done. Good work there, Devin. The genesis of this project really comes out of a discussion that I had with a couple of colleagues more than 18 months ago, where I think part of the investigation of mechanical thrombectomy needs to be veering towards identifying subgroups where reperfusion is futile, having a more discrete and directed approach to how we evaluate these patients and how we evaluate their imaging and their likelihood of having a good outcome. And obviously the sample size is too small to really delve into that too much. But as Devin said, I think there is something to be gained out of larger population evaluations, multi-center evaluations, and looking at patients pre-interventional, pre-selection imaging with far greater detail and subsetting the aspect score in the previous stroke risk to outcomes for these patients.
Okay, thank you very much. We'll move on to Ray. Ray, I see that your video might not be working. Is your audio working?
You hear me?
Yes, I can hear you.
Okay, so yeah, I'm not sure why my video is not working, but as long as everybody can hear me.
I'm going to share your slides.
Mm-hmm. Okay. Can everybody see my slides? Okay, perfect. Okay, Dr. Kellner, you can see my slides, right?
Yep, go ahead.
Okay. Okay, all right. Good morning, everybody. My name is Ray Fang. I'm a second-year resident at Mount Sinai Neurosurgery, and today I will be talking to everybody about this neurosurgery selection process. And I titled my talk this way is because I have an interest in this topic from my psychology background, and also in light of the current situation where sub-Is are being canceled, interviews are being moved to a video format. I think there's a lot of opportunities for intervention and improvement of our current process. So why do we care about resident selection? Well, neurosurgery residency is this unique position. It's very demanding. It has long hours. We have to have the mental fortitude to deal with high acuity diseases and to be able to manage and recover from devastating complications. We also have a very unique team dynamic because our programs are so small. And another thing is research have demonstrated that neurosurgery residents have a distinctive personality type. The Myers-Briggs type indicator is a very commonly used personality typing indicator that's been utilized in corporate and industrial world. And as people can see, the scientists and the executive subtypes takes over half of neurosurgery residents. However, both of these types combined is only more than 3% in the general population. So this indicates that we're a very unique group of people. But on the other hand, we also care about selection is because the cost of ineffective selection is very expensive. I've given you some data on this slide about the cost of remediation and the attrition. The more significant impact about attrition is that not only it has monetary costs, but it also gives a negative impact on the reputation of the program, team morale, and can be very difficult to recover. Also, I'm sure everyone have experienced the scramble and the stress to go through where a very small team have to cover additional job burden when somebody has to drop out unexpectedly. And also, ineffective selection can increase the disparity of our specialty. As everybody already know, we have a very large gender disparity, and the part of the trend is because the female attrition rate is much higher than the male attrition rate. So this leads to a lack of female mentorship in the specialty, and that makes recruitment and the retention of other talented women to join us even harder. And this just goes into a negative cycle that exacerbates itself. So what are we doing right now to select for residents? We look at pre-residency factors such as test scores. We use unstructured interviews, and the emergency medicine program actually use a standardized video interview. But like my little comic in the corner suggests, all these methods so far is about as random as musical chairs. So for pre-residency factors, essentially it has been demonstrated that no factor really predicts residency success. Test score correlate well with board scores as predicted, but it correlate poorly with faculty evaluations. Some modest predictors can include aggregated rank score, letters of recommendation. However, there's only very limited evidence-based approach in using these to predict the overall residency performance. And unstructured interviews can be even worse. They're very heavily weighted. However, they're very poorly validated. Again, there's no correlation with clinical performance, and they're very subjective and prone to unconscious biases. Inappropriate questions can even worsen racial and gender discrimination. Standardized video interview is a good step forward in trying to use something that's structured. However, this method has also not been studied longitudinally, and the current data also have not shown to be correlated with performance. And it's also very redundant with the step 2 exam that everybody has to take. What are the future directions that we can go? So the science of selection is much better developed in industries, and there has been a lot of data demonstrating that standardized questions and personality tests can correlate well with job performance, can reduce turnover, and increase job satisfaction. So commonly used methods can include structured interviews, personality assessments, and situation judgment tests. Structured interviews are much better because they're more objective. They ask only job-related questions, and everybody rates the applicant on standardized scoring format. And it has been shown to have better predictive validity and is more reliable. Personality assessment not only have been shown to predict job performance in a variety of fields, but it has also been shown to predict well in surgical fields. It has been studied in the general surgery program, in orthopedics program. And this is not to imply that one personality type is a perfect fit for neurosurgery. However, knowing one's type helps team building, helps differentiate learning styles, and self-reflection can improve leadership and management style. Situational judgment tests have also been well studied, and it's based on realistic work relevant situations. Again, it's scored in a very standardized way with a predetermined key. So there has been shown to be good validity, and also it provides the applicant a realistic job preview and encourages them to consider their own fit as well. So what are the future opportunities? Well, the anticipated changes that are coming up is that step one is becoming pass and fail. More and more interviews are going to be done online over video. So we're also more likely to receive more applications as people have less traveling conflicts and cost restrictions. So that gives a lot of additional data and opportunities for innovation. Not only can we think about using the previously more validated methods that we talked about, but it also brings up opportunities for developing AI processing of application data, social media analysis, as everybody increasingly have a more of a presence online, and also possibly assessing for applicants' surgical skills using surgical simulation technology. So in conclusion, poor selection can be very costly. Alternative methods are out there that have better validity, and we need longitudinal validation for these methods. And this is important because the future of the specialty is really dependent on how we select and train the next generation of neurosurgeons. Thank you, everybody.
May I ask a question?
Yes, Dr. Patterson.
I thought that was a fantastic presentation. I really liked every bit of it. One question I have has to do with the use of video at all. Since so much of what we judge is based on appearance, you could imagine that something like the color of someone's skin, the way they have their hair, how they dress, whether they're obese, you know, anything that might come across in the appearance on a video, which may or may not be related to performance. I'm not saying that none of it is unrelated, that all of it is unrelated, but how does that influence things? And what is the validity of the video itself?
Yeah, that's actually a very good question. I don't know if that has blinded interviews had really been studied well. I can look into that. But yeah, like in the musical world where like musicians conduct blind auditions where the evaluators only listen to their performance has really been shown to decrease racial and gender discrimination. So I think this is a very, very good point, and we should definitely look into it.
Yeah, Ray, I think that's great. Do you have a next steps vision for what you're doing and where you plan to take this work?
Yeah, I think the first step, actually, I would really like to study how our faculty evaluate people during interviews. Like, what are the questions that they ask, and what are the information and sort of the points that they try to pick up from an applicant's answer. And the next step is to implement the kind of more validated evaluation methods that we previously shown. So maybe instead of asking the applicant random questions, we can set up like a situational test. For example, asking them like, how would you talk to a family that about a patient that potentially needs an EVD. So we can assess their knowledge about getting consent for the process. We can assess their communication skills in the way that they try to talk to the family, and we can also assess their like prioritization skills in the way they the way that they bring up different points at different times. So I think I think that this will really give us more objective data and help with the selection process to be less random.
You've just been elected to do this for us, if Jay approves. But I think it's all of these are really fantastic ideas.
Absolutely.
Okay, thanks very much,
Thank
Ray.
you, sir. Thank you.
Will Schumann?
Yep, I'll share my screen. Can everyone see that?
Yes.
All right. Good morning. Thanks for having me. My name is Will Schumann. I'm a third-year med student at Sinai. I'm presenting my work with Dr. Caridi, looking at ASA status in spine surgery and associations found with length of stay and cost of care. So previously, another med student who worked with Dr. Caridi wrote a manuscript showing that ASA status is a predictor of hospital length of stay, ICU length of stay, and also direct cost for patients undergoing lumbar laminectomy and fusions. And our goal today was looking to see if these findings were also true for 4 other commonly performed spine procedures. So the reason this question is important in the first place is spine surgery rates have been increasing since the beginning of the 21st century, as well as the cost for these procedures, especially certain procedures such as spine deformity procedures. Also, as more and more systems use bundled payment models for reimbursement, it's important to know what the drivers of cost and patient outcomes are for these commonly performed procedures. So how we accomplish this, our study is using an administrative database of all spine surgeries at Sinai from 2008 to 2016. We queried for the CPT codes of these different procedure types to make our cohorts, and to make sure they were appropriate cohorts, we made relevant exclusions. So excluding acute pathologies, excluding any instrumentation or fusion in our lumbar microdiscs, excluding pediatric and adolescent patients in spine deformity to make sure we're looking at adult spine deformity, and then excluding any front back procedures in our ACDF and PCDF patients. So we compared demographics between all the cohorts and used them in multivariable regressions in order to analyze direct costs for patients, as well as length of stay and perioperative events and readmission rates. So what we found, in the interest of time, I only included a few demographics, but we can see that with age gradually increased with increasing ASA status, and the differences found between these cohorts for every surgical type was significant, as indicated by the highlighted p-values. And we also looked at number of segments fused in order to assess the invasiveness of surgery. And so what this table is telling us is that spine deformity patients were undergoing similar invasiveness of their surgery, regardless of their ASA status. And the cervical patients, although the median and the interquartile range, which is what we're showing here, may not fully capture the distribution, what our significant p-values are telling us is that the distribution of the number of segments fused varied based on the ASA status. So for ACDF patients, it looked like ASA 4 patients had maybe slightly fewer segments fused, 2.5 median instead of 3. And then for PCDF, maybe the higher ASA status patients had some more, third quartile of 5 instead of 4. And so for looking at direct cost for our outcomes, we use a linear regression controlling for several variables. And what this table is showing us is, again, direct cost gradually increased with ASA status. And high ASA class had significantly, patients with high ASA had significantly higher cost of care than their reference groups. So a microdisc patient with an ASA status of 3 on average had a direct cost of $1,100 more than ASA status 1 patient undergoing the same procedure. You see that in spine deformity and PDGF, we grouped their ASA statuses into high and low classes. So high 3 and 4 ASA versus ASAs of 1 and 2 and low ASA class. The reason we did this was you can see that their populations were slightly smaller for these cohorts. So in order to power the study better to look at these outcomes, we grouped them into high and low, as has been done several times in the spine surgery literature. Similarly, for length of stay, we see gradually increasing length of stay with ASA status. And again, patients with high ASA class had significantly longer length of stay than patients with low ASA class, and patients with an ASA of 2 had similar length of stay to patients with an ASA of 1. Again, in the interest of time, I only included significant findings for our perioperative events. And what we found was that these events varied by surgery type as far as their association with ASA status. And this was done with a logistic regression. So what this is telling us is for each unit change in ASA status, for every 1 level change, the likelihood of this event occurring was to increase by this odds ratio number. So a microdisc patient with an ASA status of 2 is 11 times more likely to require an ICU stay than a microdisc patient with an ASA status of 1, and so on and so forth. So an ASA 3 patient would be 11 times more likely than an ASA 2 patient. And the reason that I highlighted required ICU stay and non-home discharge, as you can see in these 2 slides, is it was significantly associated with ASA status in all 4 surgery types. So the other events varied as far as their association with ASA status, but required ICU stay and non-home discharge was associated in all 4 surgery types. So what we can conclude with these results is that ASA status is a predictor of not only length of stay and direct cost for our spine surgery patients, but also of ICU stay and non-home discharge for all these several types listed. Other operative event or perioperative events are also associated depending on the surgical type that the patients underwent. And the value for this is not only in having discussions with patients and creating care plans while they're in the hospital, but also because we were able to quantify the outcomes as far as length of stay and direct costs, it can be useful in forming healthcare policy as more and more episode-based models are used in healthcare. And I'd just like to thank all my mentors and teammates, Dr. Caridi and his group, as well as Dr. Steinberger, Dr. Cho, and Dr. Gall in anesthesia. And of course, all the residents and other med students who've helped me make these projects possible.
John, Jeremy, any comments?
So it's a little intuitive that, you know, sicker patients are going to cost more, but it's never really been shown before. And I think, you know, by doing it in the separate populations, you know, doing it for different types of surgery, as we show that, you know, a sicker patient, you know, is going to cost more, it's going to benefit us once the episode-based payments get put into place. So, you know, that's our goal in doing a lot of these studies, is to show that it's not that patient factors do contribute significantly to their overall costs and length of stay.
Nice job, Will. Kostas here. So, you know, I think this is great work, and it kind of goes along with the story you're trying to build with Dr. Caridi, that more complex patients require more resources, and therefore, you know, there's a strong argument to get more reimbursement. So I guess the question is, you know, when these types of things are negotiated, this type of data is important. And I just want to see how we envision bringing that forth outside of publications. I mean, I think this is something that needs to be kind of showcased to our leadership as they look at contract negotiations with insurance companies?
Yeah, I think it's definitely valuable for that. The benefit of ASA status is every surgical patient has an ASA status, and it's a one number that can widely be used for all surgical patients. And it's interesting to see how it can be used maybe with or versus different frailty scores that have been mentioned today.
Okay, thank you very much, Will.
Thank you.
Abi, let's try to screen share. I know Abi's sharing from an iPad. I'm not sure that's going to work, but give it a shot. If not, I have your slides.
It should work now. Give it a second.
We do see your
Are you
screen.
able to see my screen?
Yes, we can. But we see, there we go. Good.
Hi, good morning, everyone. I'm Abi. I'm one of the PGY1 residents here at our department. I'm going to present on the topic of surgical management for prolactinomas. The faculty sponsor is Dr. Kellner. This is a national overview regarding prolactinomas. So I don't have any disclosures regarding the study. So a little bit of a brief overview. Prolactinomas are the most common hormone secreting, pituitary tumor. They comprise 50% of all pituitary adenomas and 2% of all intracranial tumors. Usually presented, very rarely presented as incidental findings, but most of the time presented having clinical manifestations such as headaches, visual compromise, especially for macroadenomas and giant prolactinomas, delayed puberty, amenorrhea, gynecomastia, and galactorrhea. And these symptoms vary based on the gender of the patient presenting. To this day, the treatment for prolactinomas initially used to be the surgical resection was one of the mainstay treatments, but given the advances in dopamine agonists with progressively less and less long-term side effects, it has since replaced surgery as a first-line treatment for most patients. One of the benefits is that it normalizes prolactin levels and significantly reduces tumor size, up to 80% in a lot of the cases. However, surgery is still reserved for patients who do fail medical management, and also for patients who have malignant prolactinomas, that are extremely resistant. And also, there are a few cases where patients are looking for more of a quote-unquote permanent cure, and they want to avoid the long-term effects of dopamine agonists because either they're one of the small groups of people who are extremely sensitive to those agents, and it's affecting their quality of life. So they choose to take the risk early on, as opposed to continuing therapy for the rest of their life at a higher dose of dopamine agonists. Radiotherapy, just to mention it, it's not commonly used, but it is an option for prolactinomas that are resistant to medical management as well as surgery. Focusing on the gross total resection of prolactinomas, since medical management can significantly reduce the size of most of these tumors, I really was not able to find any literature that was directly evaluating the relationship between the size of the prolactinomas on diagnosis and the probability of the patient requiring a surgical option, specifically gross total resection. Because there are cases where patients do undergo biopsy and then are treated with dopamine agonists. But I wanted to focus more on a tumor that requires a full resection based on the initial presentation. And the reason I think that it's not really clear-cut for prolactinomas, because for other adenomas and non-secreting adenomas who do not shrink significantly with medical therapy as much as prolactinomas, it's easier to assume for those tumors that the larger tumor will more likely require surgery. but it's hard to assume that for prolactinomas due to the fact that they can be significantly shrunk by medical management. So the study design, we used we retrospectively reviewed a multi-institutional database from National Cancer Institute's Surveillance Epidemiology and Results Program, included 18 cancer registries, and patients were analyzed from 2004 to 2016 with a diagnosis of prolactinomas, so excluding all other types of pituitary tumors. The methods of the study, we analyzed the demographical and treatment characteristics.
It looks like there's something weird going on with the slides where they're overlapping. I'm not sure what's going on, so I'm just going to share from my screen, okay?
Okay.
Okay. Okay, go ahead.
All right. Sorry. Thank you, Dr. Kellner. So the methods included analyzing demographical and treatment characteristics of these tumors, ranging from 1 millimeter to 70 millimeters in size. We also use multivariate logistical regression to analyze the probability that the patients who will undergo post-total resection compared to medical management only. And just as a secondary outcome, use a Kaplan, because SEER has a survival data available for the patients, we use a Kaplan-Meier curve with log rank test to plot overall survival between the medical management group versus the gross total resection group. So just overview of the results. Stratified initially, the group was stratified into micro prolactinomas and macro prolactinomas, 1 to 9 millimeters, and I included giant prolactinomas in the macro group because there isn't really a clear-cut basis for those two distinctions, besides an arbitrary cutoff at 40. So as you can see, most of the patients are diagnosed between the ages of 21 to 40, where an interesting thing is the macro prolactinomas are diagnosed much more frequently in older patients between 61 to 80 years old. You can see 14% of those larger prolactinomas are diagnosed in this elderly group, compared to only 3% of those microadenomas. There is obviously a gender or a sex significance, with females having 60% of total prolactinomas compared to males, with a lot more microadenomas having a female predominance, which is consistent with the literature that is around. And in terms of the region, this is just encompasses the patients found in different regions in SEER database. So it kind of shows that it's not taking a homogeneous sample across the country. There are some regions of the country that are more heavily, focused, that it more heavily focuses on compared to the other regions. So they're overrepresented. So just to keep in mind when, interpreting the results. Okay, so this is a figure that shows tumors stratified by 10 millimeters of size, comparing gross total resection versus medical management. And as you can see, it initially clearly shows that the gross total resection, the percent of the patients getting gross total resection for these prolactinomas increases almost linearly up to 30 to 40 millimeters. After that, it kind of flattens out until 60 millimeters in size. And that's why it didn't really make sense to, initially group a giant prolactinoma as separate from macro, because that cutoff really doesn't mean much at 40 millimeters.
Have you just a minute left?
All right, so just final results, multivariate logistical regression. It's comparing the patient probability of the patients, which is showing, as you can see, patient compared to the patients with microprolactinomas. As you get larger in size, you get threefold, fivefold, and eightfold increase in the probability of undergoing surgical resection, even after controlling for age and sex. As you can see, if you go all the way to 40 and 50 millimeters, your chance of undergoing surgical resection are 8 times more likely. And this is the first time it has been shown for prolactinoma specifically that surgical GTR is influenced by initial size. This is just a survival curve showing no significant survival difference between the two groups. So in conclusion, obviously, this is to my knowledge, the first study to observe a relationship between the size of prolactinoma and diagnosis and the odds of undergoing post-total resection. And as even though the role of surgery is not as significant as it was, 30, 40 years ago, it's important to know that it does play an important role in management of these larger prolactinomas. And the 60% female predominance is consistent with a lot of information found in literature. And as a secondary outcome, there was no real difference between overall mortality on these patients who underwent gross total resection versus medical management.
Can I make a comment?
Yes.
So it's an interesting study you're doing. This is Dr. Post. One needs to be a little cautious in talking about gross total removal, particularly with the large tumors, defining gross total removal by both MRI imaging as well as prolactin levels. While it may look at times like the scan is clean, if the prolactin isn't normalized in a lot of the individuals, you can't count it as a gross total removal, and they still do need medication. It's not surprising that it's less symptomatic in older individuals because they're out of childbearing years for the women, and the men are just less sensitive to it. We tend to see the larger prolactinomas much more so in men than in women. And even for the small tumors, that when you look at the studies that are around, you have to consider what the long-term remission, or using the term cure, I don't like to use the term cure until 10 or 20 years down the road, what the remission rate is, and then the lack of need for dopamine agonists in addition. A lot of people are doing studies trying to show that the cost of long-term treatment is less with surgery than it is with long-term medication. That's presuming that you have the long-term remission. And the study out of Austria, out of Vienna, showing that even for the microadenomas, if they're lateral in the stellar, they only had a 55% long-term remission rate because of the involvement of the cavernous sinus wall. But the bottom line, these studies are interesting and it'll always get revisited. Prolactin was one of the earliest, it's the first paper I wrote on pituitary tumors on the success of resection. And it holds at times. A nice study.
Thank you.
Okay, let's move on to Emily.
You can see my video, right?
Yes, we can.
Okay. Can you see my screen?
Yes.
Great. Now it's a presentation view, right? Okay. All right. Hi, everybody. My name is Emily Chapman. I'm a third-year medical student here at Mount Sinai. I'm about to embark on a scholarly year and with the department. And today I'm going to speak to you all about dysplastic cerebellar gangliocytoma, also known as DCG. It's a rare brain tumor, and the inspiration for this project was with a case, one of Dr. Germano's recent cases. We decided to look further into the literature and also into cases at this institution. So no relevant disclosures for this presentation. And let me launch right into DCG. So again, it's a rare ganglion cell cerebellar tumor. It was first described basically exactly a century ago. And again, since there are not many cases in the literature, our gradual understanding has culminated in 30 years ago. A group, Padberg and colleagues, recognized it as pathognomonic for Cowden syndrome. Cowden syndrome is an autosomal dominated genetic syndrome. It's characterized by a PTEN gene mutation. As actually discussed in our keynote lecture earlier this morning, PTEN is a really important driver in many different cancers, and it has also been characterized in CS. Patients are at increased risk of multiple hamartomas throughout the body, as well as various cancers listed here. To this day, the nature of DCG in particular is controversial. 4 years ago, the WHO assigned it low grade and mixed neuronal glial tumor category. So again, in this study, we're really looking at the literature and some cases here in the institution to better understand and characterize this tumor. We did a lit review in PubMed over the past 5 decades. We also did an institution search over the past 20 years. We extracted data and we divided into adult and pediatric cohorts and collected a bunch of demographic, diagnostic, and treatment variables, which I'll get into soon. This is a Prisma diagram, pretty standard for meta-analysis. You'll see that actually the number of citations in the literature at first. We went through all the different sources and ultimately came up with 41 citations for 5 case series and 36 case reports. These are the cases in literature that we really worked with. Some initial demographic results here, a total of 111 cases across all these sources in the literature. But you'll see here there's great variability, right? So the first 15 years, there were no sources that we were able to find. And then almost half of all cases really in the past 20 years. We attribute this to really the advent of MRI, which I'll explain is pretty important for the diagnosis of this tumor. And that really informed when we looked in the EMR at Sinai, really looking in the past 20 years at the cases. Another important point here is that the tumor remains predominantly among adults. This was also reflected in our institution cohort, which is particularly interesting given that, as I mentioned, it's an inherited disorder. Some more demographics here. So among the literature and our institution cohort, we saw a kind of a middle age, middle age, between anywhere between 40 and 65. The pediatric cohort, there was much greater variability. We saw in the literature, we saw neonates, we saw all the way up to age 20, which was really the cutoff for our pediatric cohort. In terms of gender, a slight majority of female patients, both in pediatric and adult cohorts. And although the sample size for our institution is still small, that's also what we saw in our cohort here. Moving on to the diagnostic characteristics. Again, T2-weighted MRI is really standard for diagnosis. And the catchphrase here is tiger stripe pattern. You'll see in figure A, it's this alternating strided pattern of both hypo and hyperintensity on T2-weighted MRI. Again, we really looked at the past 20 years in the literature and saw that this literal explicit tiger stripe pattern was mentioned in a good number of these cases for both adult and pediatric. These images here are from one of our patients. In B and C, you'll see the tumor itself. You'll see mass effect and tonsillar herniation leading to hydrocephalus, which really highlights the symptom presentation, both cerebellar and hydrocephalus symptoms in the patients here. Also important for diagnosis is histology. In this wider magnification view, you'll see a disruption in the granular layers of the cerebellum disrupted by these dysplastic ganglion cells. In the higher mag view, you'll see the disrupted the dysplastic cells themselves with the arrows. And really, this disruption in the layers is also again correlated with that alternating strided pattern that I mentioned in MRI. Also, immunohistochemistry plays an important role. Neurofilament immunostaining in particular is characteristic, which is reflective of the gangliosites in this tumor. Some initial genetic analysis, again, in the more recent literature, we saw that ever since it was first mentioned in 1990, Cowden syndrome was assessed in the majority of both adult and pediatric cases. The number is a bit lower in our series. Again, we have to, again, this is a smaller sample size, but we're doing some further genetic analysis. It's been a bit of a delay on that because of the recent pandemic, but we're ramping up the genetic sequencing for the samples in our institution. And again, why do we care about the genetics in particular for CS? As I mentioned earlier, so unlike kind of multiple tumor factors like you might see in some other tumors, Cowden syndrome is really characterized by just a P10 mutation. And so we thought it's an interesting human model in the sense that there is one mutation. We see incomplete penetrance despite it being autosomal dominant. And we think that this reflects a spectrum of presentation likely due to epigenetic factors. So we're really interested to see how these epigenetic mechanisms might impact the expression of this one gene and therefore the presentation of this tumor. In terms of outcome, what's great, luckily most patients have good outcome. In the literature, we saw that 90% had favorable outcome. There were 2 patients that needed TMZ for progression. The tumor margins are often difficult to appreciate intraoperatively because these dysplastic cells tend to blend in with the surrounding parenchyma. But you'll see in these images from our institution that there's great resection post-op. So this brings me to the end of my talk. Again, this is a quick summary. Again, I want to emphasize the importance here of including probands that have been diagnosed with CS, but also people who aren't showing symptoms but have a mutation. I think this will be really key to better understand the full spectrum of expression, like I mentioned, of this one mutation. There's a lot of diagnostic complexity surrounding CS. Like I mentioned, there's tumors throughout the body, so it really highlights the importance of collaborating with a variety of specialties to best treat these patients. We're really excited to see some more whole exome and whole genome sequencing, both in our cohort and also in the wider literature. And again, resection remains the gold standard and it has provided a good outcome. So that's the end of my talk. I really wanted to have some acknowledgements here. I really wanted to thank Dr. Germano for her guidance with the literature and also with the cohorts here. And also Dr. Bryce from the Department of Pathology. She provided all the histopathology and genetic data. And I finally wanted to thank the department for this opportunity to present. Thank you so much.
Emily, this is Isabel Germano. Congratulations. Great talk. I also wanted to congratulate you for being part of the Scholar Award for the next year. Emily has worked really hard over the past year. I was her mentor and she put together a very nice proposal. She will be working primarily with Dr. Mocco. She has an incredible amount of energy and as some of you know, besides working with me, she was also able to put together projects in cerebrovascular and spine. So it's really nice to have you in the department for the next year. Thank you, Emily.
Thank you.
Okay, next speaker is Ernest.
Good morning. There we are. Okay. Are you seeing this in presentation?
That looks good.
Okay, great. So good morning, everyone. I'm Ernest Bartholomew. I'm a PGY6 resident, and I'm presenting this talk on behalf of Alex Schuper, who we heard from earlier this morning. He's one of my junior residents. And he, as well as Tom DiChiara, a research assistant over at Elmhurst, did a significant amount of work on this project, looking at the effect of alcohol intoxication on TBI outcomes. So with that, none of us have any disclosures to make regarding this presentation. So the basic science literature has suggested in a selection of studies that there is a potential neuroprotective effect of ethanol in the context of TBI. This has panned out inconsistently in clinical studies. There's actually a lot of controversy and a lack of consensus regarding this effect. So we sought to investigate the relationship between acute alcohol intoxication at the time of injury and outcomes following traumatic brain injury for patients presenting to Elmhurst, which is a level 1 trauma center. And we did this hypothesizing that alcohol intoxication would actually worsen outcomes. And this was based on some anecdotal observations that perhaps due to platelet dysfunction, alcohol intoxicated patients had worsening hemorrhages after traumatic brain injury. So At our center, we performed a retrospective analysis, looked at 5 years of data from 2014 to 2018. And our inclusion criteria included patients with acute traumatic brain injury who were adults, had an abbreviated injury head score, head injury score of greater than or equal to 3. So that ranges from serious to critical or maximally injured head injury versus an abbreviated injury score of less than 3 for the body, so mild or moderate. So we're trying to minimize the impact of non-brain injury on these outcomes, as well as documented blood alcohol level on admission. So we excluded patients who had prior brain injury, anyone with evidence of coagulopathy, INR greater than 1.6, thrombocytopenia, et cetera. And then we performed univariate and multivariate statistics to assess the effect of alcohol intoxication on our primary outcome, which was in-hospital mortality, as well as the secondary outcomes that you see here. So in the way of results, and we'll just go through these and then I'll present our data tables, 527 patients met our criteria during the period of study. And they were divided into 2 cohorts, one with positive blood alcohol level, 267, and then a negative blood alcohol level in 260 patients. In terms of baseline characteristics, they did differ by age. The blood alcohol level positive patients were younger, and they were also predominantly male. However, there was no difference in terms of severity of traumatic brain injury among the cohorts. And essentially, we found that our hypothesis was not supported by this. So mortality at discharge was 10% for the blood alcohol level positive patients versus 9.2, and this was not statistically significant. That was our primary outcome. Also, no significant difference in terms of secondary outcomes, and this is regardless of controlling for potential confounders here. So here's our first data table demonstrating the baseline demographic characteristics. And as I mentioned, the blood alcohol level positive patients were younger and predominantly male, but no other statistically significant difference was observed, not in injury severity score, not in the discharge Glasgow coma score, or even length of stay. And in terms of the rest of our analyses, so the main differences we saw are differences that we can predict based on other variables. So, so those who survived were likelier to survive if they were younger. They were likelier to survive if they had a better presenting Glasgow coma score, as well as a better injury severity score. So alcohol intoxication in this group did not appear to have any significant bearing on the primary outcome, which was discharge mortality. So we found no association between acute alcohol intoxication or absolute blood alcohol level on discharge outcome. And one of the strengths of this study is our N. So it's a large number of TBI patients who were admitted at our level 1 trauma center. We have understood limitations such as the retrospective study design. However, this is part of a larger project where we're trying to better understand this relationship. We do plan to conduct a systematic review and meta-analysis of this topic, as well as see whether we can tease out what's happening in terms of these results. So with that, I'm happy to turn this to any comments that Dr. Hickman has and questions.
Thanks, Ernest. As Ernest mentioned, this is, we decided to look at our own patient population, but we're also simultaneously conducting a systematic review and meta-analysis, and we're going to include our data in that. Basically, in the literature, about half of them have shown, half of the published studies have shown no difference, and of the other half, they're evenly divided between neuroprotective and deleterious effects. One of the things we're adding to this study going forward for a manuscript is that isn't included in any of those studies is really a rigorous review of all the CT data, including these patients, which you can imagine is taking some time with 500 and something patients to include all of the CT data from admission. And over their first 72 hours. As well, we controlled for a lot of the potential confounders in terms of patients that might have sequelae of chronic alcohol abuse, such as thrombocytopenia and coagulopathy, which could affect outcome. We really wanted to look at just the effect of the acute intoxication, and really other studies haven't really controlled for those. So we're going to look at some secondary outcomes in terms of hemorrhage expansion and need for OR, but at least in terms of the impact on the short-term outcome, it doesn't seem like there's an effect in our cohort anyway.
I have a question. This is Dolores. A very nice talk, Ernest. And my question is, if I may not understood this well, so you did the study only during the admission, you checked if they are intoxicated or no. So do you happen to look at the history of seeing if they chronically use it, or it's only one time point of being yes or no for intoxication and correlation with TBI?
Yeah. Sorry.
That's good.
Go ahead, Ernest.
Right. So, I mean, in this particular study, we didn't control for history so long as the history didn't produce any other of the coagulopathic factors that were part of our exclusion criteria. So we were really looking at on admission, what was the blood alcohol level.
Okay, cool. Thank you.
How about thymine deficiency? So people who are deficient in thymine, they have some kind of overlapping symptoms of psychosis, which may be a confounder for having head injury associated with this. So did you look into that side as well?
So that's a really interesting question. And there's a number of things we could look at. We didn't, in this particular study, look at whether thiamine deficiency was a confounder here. In terms of the questions that we'll look at, first of all, in terms of what could be going on with this data, I wonder whether we have both the neuroprotective and deleterious effects happening at the same time. So, some of the basic science research is suggesting that maybe there are effects on BCL2, and so that's affecting apoptosis, or perhaps there are immunomodulatory effects, or a change in aquaporin expression that is affecting rainwater, and whether that's somehow improving the outcomes, while maybe there is platelet dysfunction. So there's a number. This is a very large project, and I think we'll certainly consider looking at thiamine and withdrawal symptoms, and perhaps how that fits into this picture as well.
Yeah, obviously we're limited because it's retrospective, so there's some data that we don't have, like we don't have thiamine levels, obviously, and if we had platelet function levels on all these patients, that would be also good data to have. I've only seen actually one study that ever really looked at coagulopathy numbers based on TAG or platelet function in these patients. In some basic science literature, there's some indication that acute alcohol intoxication does affect platelet function. And I think we all anecdotally see that when we were operating in patients, but it's been looked at in TBI and there's been no apparent difference. But again, that was only one study. So yeah, I think it's an interesting question, but I think there's probably multifactorial issues going on.
Okay, thanks very much. Let's move on to the next speaker. Next up is Trevor.
Hi, can you guys hear me?
Yes.
Great. Let me just go ahead and try and share my screen here. Okay. Hi, good morning, everybody. My name is Trevor Hardigan. I'm a PGY2 in the Department of Neurosurgery here at Mount Sinai, and I'm going to be presenting on some research that I've been doing in association with Dr. Kellner, looking at the association of posterior fossa AVMs with prenatal aneurysms and associated functional outcomes. We have no disclosures that are related to this current work among any of the authors. So posterior fossa AVMs, typically when we're looking at them, a rupture of a posterior fossa AVM has been associated with a higher risk for significant morbidity and mortality than those found in the supratentorial compartment. Research has shown that 84% of these posterior fossa AVMs will actually have a hemorrhage on presentation. 14 patients in a study demonstrated that they had an annual rupture rate ranging from 7.5 to almost 12% in the first 5 years, which was greater in this particular study than that found in the supratentorial AVMs. About 9% of intracranial AVMs are associated with either intranidal or flow-related prenatal aneurysms. This is thought to be due to hyperdynamics in the feeding artery, which can also predispose to increased shear stress and then breakdown of the medial wall and subsequent development of aneurysms. The flow-related prenatal aneurysms also have been shown to occur more commonly with these posterior fossa AVMs compared to supratentorial AVMs. Nissen and Lawton recently have proposed a new grading system for cerebellar AVMs that focused on certain factors that they felt predisposed to worse functional outcomes. These namely were the need for emergency surgery, venous drainage, specifically deep venous drainage, preoperative neurological status. Understandably, if someone is worse at baseline, then they would likely not have as much reserve to handle such an insult, as well as advanced age. The association between prenatal aneurysms and posterior fossa AVMs and the functional outcomes, however, still remain an under-examined area in the treatment of this overall patient population. Previous studies either looked at just all posterior fossa AVMs, whether or not they had a prenatal aneurysm, or they had studies focused on looking at characterization where a actual resection or obliteration of the AVM was not necessarily included, and thus the outcomes on functional status were less robust. For our study, we looked at an ambidirect review of all posterior fossa AVMs identified during endovascular procedures in our institution from July of 2016 until June of this year. The patients included in the study were determined based off of neuroradiology reports and op notes that mentioned the presence of not just a posterior fossa AVM, but also a prenatal aneurysm. The functional outcomes for the cohort of patients were looked at as an MRS at time of discharge and 30 days after discharge. Favorable outcomes were defined as MRS 0 to 2. We looked at a multitude of characteristics associated with these patients. So our patient average age was 52. We had 21 patients enrolled in the study. We had a male to female ratio of 8 to 13, demonstrating some female predominance, which agrees with some of the other literature looking at these posterior fossa AVMs. 71% of the patients actually presented with a hemorrhage. 63 of them had an EVD placed, and 42% went for emergency surgery. The hemorrhage type of the patients was also something that we looked at. Typically, the ruptures associated with the prenatal aneurysms lead to specifically subarachnoid and intraventricular hemorrhage. And so trying to tease apart the contribution of AVM bleeds versus prenatal aneurysm ruptures was something that we also looked at. So you can see that one patient had only subarachnoid hemorrhage, 3 patients had IVH only. And then we had a mixed picture of subarachnoid hemorrhage and intraventricular hemorrhage in 2 patients, intraventricular hemorrhage with intraparenchymal bleed in 4. Subarachnoid hemorrhage as well as intraparenchymal hemorrhage was not a characteristic that we observed in any of our patients. And then the most common presentation that we saw was bleeding in the subarachnoid space as well as intraventricular hemorrhage and intraparenchymal hemorrhage. This is a particular case example that was very to many of the patients that we had. There was a 61-year-old patient that presented with a cerebellar hemorrhage, as you can see here, secondary to a found pica aneurysm. The AVM you'll see here, he had bilateral EVDs placed to deal with the associated ventricular hemorrhage, and then went for an emergency shock and decompression, after which went for an angiogram, which demonstrated the cerebellar AVM as well as an associated pica aneurysm on AP and lateral angio here. Patient ended up having the aneurysm treated first with onyx embolization, and then several months later came back with Dr. Betterson for resection of the AVM for a repeat SOC, showing total occlusion of the prenatal aneurysm and then also complete resection of the AVM. Our AVM was namely located in the cerebellar hemisphere. The average size of the AVM in the longest axis was 21 millimeters. Typically, these patients were all Spetzler-Martin grade 1. 15 out of our 21 patients met that criteria, with 3 being grade 2 and 3 being grade 3. Almost all of them were associated with superficial venous drainage. And in our patient population, contrary to some other studies, we had a much more aggressive total resection or obliteration result of the AVMs, and we demonstrated that in 76% of our patients. For the prenatal aneurysms, we identified 26 separate prenatal aneurysms, the most common being pica with 16, followed by the superior cerebellar artery prenatal aneurysms, the average size 5.7. And the majority of these were treated endovascularly, 15 with 87 having a Raymond-Roy score 1 with total occlusion and 13 having a score of 2. This is showing our MRS data. So you can see here the pre-MRS data showing the majority of patients had almost no deficits, with one patient having an MRS score of 4. After the event and then at discharge, you can see a dramatic reduction in the quality of the functional status of the patients. We had a multitude of patients that were either MRS anywhere from 3, 4, 5, or 6 compared to their pre-insult baseline. One of the interesting things that we saw was that 30 days after discharge, we actually did notice a very impressive trend towards improvement in functional status, where our favorable outcomes increased in our groups of MRS 0, MRS 1, and MRS 2 compared to their discharge MRN. So the current study is looking at namely prenatal aneurysms and posterior fossa AVMs, and this is actually one of the highest single study case numbers of prenatal pica aneurysms in particular, with posterior fossa AVMs found in the literature. The pica aneurysms account for 53% of posterior fossa ruptures in the ISAT trial. Now that wasn't necessarily associated with AVMs, but it does show a propensity for these particular aneurysms to, at baseline, have a risk of hemorrhage. The functional status in our patients, as I said, significantly worsened by discharge, although there was some demonstrable improvement. The favorable outcome at discharge, as I said, was 33% in patients versus a improvement to 54% at 30 days. One of the things that I think we demonstrated in our patients was that they typically were being treated endovascularly, such that the prenatal aneurysm was embolized prior to the AVM resection. This has been discussed in terms of a treatment paradigm, whether it's more important to treat the AVM and thus try and reduce feeding artery flow as the nidus of the aneurysm predisposing to that is removed. However, the normal perfusion pressure breakthrough theory is something that suggests that the feeding arteries and arteries to the normal brain tissue due to the AVM lose their capacity to autoregulate, such that if you remove the AVM first, then you're going to end up having an increased risk of rupture through these prenatal aneurysms that you did not treat first. And so in the majority of cases, we actually did treat our patients with endovascular embolization prior to AVM resection at a later time point. In conclusion, these posterior fossa AVMs and prenatal aneurysm associations have a high propensity to rupture and then lead to a significant morbidity and mortality. One of the things that we'd like to do is extend out our window for follow-ups to determine functional outcomes at much later time points to see if that improvement that we noticed at 30 days does continue. Some of the limitations of the study are that it is a retrospective analysis. There's sampling bias because we're a single center study. There's also some attrition bias. Our 30-day discharge sample, we have one patient that was lost to follow-up and one patient that's still awaiting their 30-day post-discharge follow-up. Also, variations in the treatment paradigms and timing, namely the timing of the resection of the AVM may have an impact on functional outcomes. Some patients were treated emergently, some patients were treated semi-urgently, and then the other ones were treated months down the road. Certain references, and I'd be happy to take any questions.
Thanks a lot, Trevor. We have time for one question. If there's anybody. How do you feel SRS plays a role here? I know that some of these patients got SRS, and what plays into it?
It will play into, as I said, I think it was one of the limitations of our study thus far that we have to examine the treatments that the patients received. The number of patients in this study, while higher than in a lot of other studies examining this topic, is still small. And so for patients that received SRS, which I believe was only a couple in this study, to determine how they did as a result of the treatment of the aneurysm, the prenatal aneurysm that had as we've demonstrated, had a higher propensity to rupture versus the AVM itself, it's something that we'll have to continue to look at. The overall theory for a lot of these is that the pica aneurysms in particular, which we had the most of in our study compared to SEA or Icahn, it is felt that they are the predominant reason that these patients have hemorrhages. The AVM itself is felt to be less likely the culprit for rupture, and so SRS of the AVM perhaps might help with some of the propensity for the pica aneurysms to rupture, but still needs to be determined further.
Thanks a lot, Trevor. Adam Lee.
Yep. Can everyone hear me and see my screen?
Yes.
Okay, great. Hi, everyone. My name is Adam Lee. I'm a third-year student working with Dr. Chowdhury. Thank you to the Neurosurgery Department and organizers for allowing me to present. Thank you to the co-authors for working with me. Today, I'm going to be talking about high altitude's effects on concussion. I have no disclosures, but Dr. Mark Lovell is the co-founder of Impact Applications. So many previous studies have been performed on concussion, but much is still unknown about factors that affect concussion incidence, one being high altitude. So 6 prior studies have been performed on high altitude and concussion incidence. None have been performed on concussion severity or recovery. 2 have been by our group, and 4 show a decrease in incidence at high altitude, which is interesting given that I would think that there would be an increased incidence at high altitude. Additionally, prior studies have been criticized for using low cutoffs for high altitude, all below 1,000 feet. In this figure from a review, authors point out that physiologic changes don't occur until 2,000 or 3,000 feet. And with the exception of some locations in the country like Denver, Colorado, most, including those in prior studies, fall below this threshold. So some studies finding decreased incidence at high altitude attributed to the slosh effect, where increased cerebral blood flow at altitude causes decreased brain movement during head trauma. Interestingly, some studies have hypothesized that jugular vein compression is a potential way to induce the slosh effect in athletes. And today, athletes at all levels, from the amateur to professional level, are trying to protect themselves against concussions using jugular vein compression. We want to know, is this effect real? Does high altitude truly affect concussion incidence, severity, and recovery? Additionally, prior estimations of concussion severity have only used binary markers. We need to develop a better methodology to grade concussion severity, which we were able to do using impact testing. Impact is the most widely used computerized concussion test in the country. It's FDA approved. It uses a neuropsychometric battery of tests to generate 5 composite scores. Here, and you're deemed to have a concussion if you have elevated scores in at least 2 out of 5 of these scores. So we recently used impact testing to develop severity index, published last month in JNS Pediatrics. We found that we could generate a score severity index by summing significant differences in the 5 impact composite scores to create one number. We found that increased severity index directly correlates with prolonged recovery, as you can see on Kaplan-Meier analysis, with the less severe injuries in blue and the most severe injuries in purple. And severity index also directly correlates with increased post-concussive symptoms in the migraine group, cognition group, sleep, and neuropsychiatric groups. Thus, we believe that severity index is a good measure for concussion severity. For our altitude study, our study population had 25,000 impact tests for about 9,200 individuals, a population larger than most studies in the literature. This is over a 10-year period. So athletes were in multiple institutions in Colorado, our high altitude location, and Florida, our low altitude location. We believe we can better study the effects of altitude here because Colorado is above previous physiologic thresholds mentioned. Additionally, standardized protocols were used for all athletes, including preseason baseline testing, head injury assessment on the field, and post-injury impact testing. Some patient demographics briefly. As you can see by the highlighted variables, there are many significant demographic variables that we're going to need to account for in multivariate analysis between low and high altitude. Starting with unadjusted univariate analysis of concussion incidence and severity between low and high altitude, incidence is on the left with our high altitude groups in black. As you can see, the raw rates of clinical head injury is significantly higher at high altitude, which contrasts prior studies. But if you only look at concussions on impact test, the trend switches with a lower incidence at high altitude. Looking at severity on the right by severity index for both the clinical head injury group and concussion on impact group, high altitude athletes showed significantly lower severity of injury. Now, looking at clinical head injury versus concussion on impact, regardless of altitude, using two-way ANOVA, we saw that concussion on impact test group showed significantly higher severity injury. From this, we think that high altitude athletes have an increased incidence of injury, but less severe injuries, and that concussion, the impact test is better at detecting more severe injuries than less severe injuries. Now moving to multivariate analysis to control for the differences in demographic variables, high altitude is on the top. This is for head injury incidence. As you can see, high altitude remains significantly associated with increased odds of having a concussion. Moving to multivariate analysis for concussion severity, with high altitude on the top again, all head injuries are on the left, only concussions on impact test are on the right. Once again, high altitude remains significantly associated with decreased odds of having a severe injury. We did an unadjusted analysis, Kaplan-Meier analysis for concussion recovery between low and high altitude. We split athletes up into 3 groups, a low severity group with severity index below 6, an intermediate to the low altitude group in black. For conclusions in our study, we found that concussion severity can be estimated using severity index. There's increased incidence of head injury at high altitude, but decreased severity of injury at high altitude. For severe injuries in unadjusted analysis, concussion recovery was prolonged at high altitude. Thus, given these conflicting results, the relationship between altitude and concussion appears to be complex. It may explain why prior studies have had conflicting results. Some limitations for our study: we only had 2 states in analysis. There are limitations to the impact test itself. Timing of post-injury follow-up was not standardized. It was based on patient availability. And there are other unknown levels of variation in participation, skill level, and other environmental factors. For future directions, we want to extend multivariate analysis to include recovery and post-concussive symptoms. We want to investigate other patient factors and their effects on concussion. We want to look at other environmental variables, and we also want to obtain additional data for more states and altitude ranges. So I'd like to thank Dr. Chowdhury for working with me on this project and leading, as well as all the authors involved in this study. And I look forward to doing more research in a productive research year. Thank you.
Thank you, Adam. So I'll make a few comments. First of all, thank you for your great work on this, and look forward to having you spend the research year. On this topic, clearly there's a lot of controversy. What are the risk factors that affect head injury? There's no question there's been experimental data to show that high altitude has a deleterious effect on animals or even beyond for certain subsets. So why is it that it may reduce concussion incidence if it does? I guess what we're showing here is it's a complex relationship. There's no question that it appears that there are some protective effects, but there are also some aspects, if you do have a severe injury, that it's worse. You know, this represents, you correctly pointed out that this is the largest one. This is by far the largest one for the altitude series. What Adam was referring to is this is one of the largest studies of all concussions that have been done. But for altitude, this is by far the largest one. And so it'll be interesting to see. This has not yet been published. It'll be interesting to see what the peer groups think. And let's see if there's any questions for Adam.
Okay, great job, Adam.
Thank you.
Next up is Alejandra.
Hi, everyone. Can you hear me okay? Okay, great. Thanks for giving me the chance to present. My name is Alejandro. I'm one of the third-year residents, and I'm going to talk to you guys about the impact of obstructive sleep apnea on the cost and outcomes in patients who underwent posterior cervical decompression infusions at our institution. I have no conflicts of interest to disclose. And so as way of introduction, obstructive sleep apnea is a pervasive and chronic condition. Its prevalence is estimated to be—
I don't see
Yes.
you're not
You don't see
screen.
Sorry about that. Do you see it there?
Perfect. Thanks.
Okay. To just give an introduction about the relationship between obstructive sleep apnea and the cervical spine, obstructive sleep apnea is a chronic condition with prevalence anywhere between 9 and 24% of the population, and it has shown to have direct correlations with all-cause mortality, as well as with other medical conditions such as hypertension, stroke, cardiovascular problems. Interestingly, obstructive sleep apnea severity has shown to correlate with different posture of the cervical spine. Specifically, patients with obstructive sleep apnea have been found to have greater head hyperextension and anteriorization. You can see a representative picture here of a patient with obstructive sleep apnea versus a healthy control. And those with obstructive sleep apnea have also been found to have more fusions and morphological deviations of their cervical spine. And although obstructive sleep apnea has been studied in terms of its effects on other types of surgeries, its effects on the cost and outcomes of posterior cervical decompressions and fusions hasn't been studied in the literature. Therefore, we hypothesized that OSA increases the cost of carrying out these posterior cervical operations and may lead to delayed extubations as well as more overall complications. We therefore carried out a retrospective review of all posterior cervical decompression and fusion cases in our institution from 2008 to 2016, found a total of almost 1,200 patients who underwent this procedure. 93 of them had obstructive sleep apnea. That's only 8% compared to 92% who didn't have sleep apnea. But we set our primary outcome as delayed extubation after leaving the operating room. And our secondary outcomes included ICU admission, any complication, an extended hospitalization, non-home discharge, and ED visits after their operation. When we carried out a univariate analysis, we found that obstructive sleep apnea correlated with higher rates of ICU admissions, as well as total complications and respiratory complications, which is consistent with literature on obstructive sleep apnea in other operations and in other settings. However, when we carried out our multivariate analysis, once again, we found that patients with obstructive sleep apnea had a significantly increased risk of ICU admission. However, there was no difference in the odds of prolonged intubation or in the total number of complications or complications by body system. Interestingly, patients with obstructive sleep apnea actually had a decreased risk of extended hospitalization, and within our cohort at least seemed to leave the hospital sooner, which is inconsistent with literature on sleep apnea in other areas. We also attempted to analyze the effect of obstructive sleep apnea on cost in patients who underwent posterior cervical surgeries, and constructed 4 different models of varying complexity, each progressively controlling for more and more variables. Within our models, we found that there was no difference in cost for patients with sleep apnea versus controls who underwent posterior cervical fusions and decompressions. I think it's important to consider a few limitations of the study. Importantly, that it's a retrospective design within a single institution, and the protocols within our institution and the way that we bundle costs may limit the generalizability of the data. We also used obstructive sleep apnea as a binary variable, knowing that obstructive sleep apnea is a complex disease with variable severity. Our sample size also included a low number of patients with obstructive sleep apnea, but from this study, we concluded that patients with sleep apnea may be more likely to be admitted to the ICU postoperatively. However, obstructive sleep apnea alone is not a risk factor for poor primary or secondary clinical outcomes following posterior cervical fusions, and it does not increase the cost of doing these procedures. And with that, I just want to thank everyone.
So I think this was an interesting study for what it didn't show, and that we were expecting that patients with OSA and cervical spine problems would have, once they had surgery, would have increased airway difficulties. But in fact, that wasn't the case. And we even theorized that they left the hospital sooner, most likely because they were diagnosed already with sleep apnea, they were on CPAP, and they were able to overcome whatever problems they had and then leave more expeditiously, as opposed to patients who may not have been, had a sleep study that showed sleep apnea and been on CPAP to begin with. So I think it's an interesting and somewhat unexpected result, but it also shows that we shouldn't necessarily be afraid to do posterior cervicals on patients who've been diagnosed with sleep apnea, because there hasn't, we've shown that they don't have a significant complication burden, more so than our controls.
Yeah, that's absolutely right. Completely agree.
Okay, thanks a lot, Alejandra. And Jeff Zimmering.
Okay, can you hear me?
Yes, we can.
Okay. Hi, my name is Jeff Zimmering, and I worked with Dr. Margettis on this project. The title of my talk is Dome Laminotomies at Adjacent Segments in Cervical Laminoplasty. So cervical laminoplasty is an established and effective surgical treatment for neurologic dysfunction associated with cervical myelopathy. Dome laminotomies involving undercutting the lamina adjacent to the laminoplasty levels to decompress and prevent both dynamic and static spinal cord kinking on the lamina edges. Now, this technique allows a decrease in the number of instrumented lamina, a smaller surgical exposure, and preservation of the muscular attachments at the top of C2 and C7. So you can see here in this illustration that was prepared by Jill Gregory, the red lines indicate undercuts at the caudal trailing edge of C2 and the cranial leading edge of C7. The result, as you can see shown here in B, is that decompression, which prevents both dynamic and static kinking of the spinal cord. Here you can see an intraoperative photograph which shows the completion of the C7 dome laminotomy that you can see here by the yellow dashed line. The blue dots show the spinous processes of C4, 5, and 6, and the yellow dot, the C7 spinous process. And this intraoperative photograph shows the completion of the C2 dome laminotomy here by the yellow dashed line. The blue dots show the spinous processes of C3, C4, and the green dot, the C2 spinous process. Now, we investigated whether dome laminotomies are associated with satisfactory neurological and pain outcomes. We performed a retrospective review of consecutive patients that were treated at Mount Sinai Hospital between November 2015 and September 2018. The patients underwent a C3 to C6 laminoplasty, including dome laminotomies of the caudal trailing edge of C2 and the cranial leading edge of C7 lamina. Post-op evaluations of pain, myelopathy symptoms, and complications occurred at both an early, mean 2-month, and late, mean 15-month time points. 21 patients met criteria for inclusion in the study, 15 men and 6 women, having a mean age of 62 years. All 21 patients had cervical spondylotic myelopathy, and 2 patients had mild ossification of the posterior longitudinal ligament. There was a mild increase in postoperative pain, which has been reported to occur following cervical laminoplasty. In our study group, 2 of the 21 patients experienced an early increase in mild pain. On the other hand, 2 of 3 patients with moderate or severe pain at baseline experienced early and late decreases in pain. Using the visual analog scale to quantify pain scores at baseline and postoperatively, we determined that mean axial pain scores decreased significantly at both the early and late post-op time points. 92% of our patients experienced resolution of baseline hand dysfunction at the early post-op follow-up, and 84% maintained it at the late follow-up. 2/3 of the patients experienced late significant improvement in their baseline balance impairment. Additionally, post-operative response rates for urinary dysfunction were 58% in the early and 42% in the late follow-ups. Image A here shows the preoperative sagittal T2 MRI, which revealed degenerative superimposed on congenital stenosis with an AP diameter as low as 4.4 millimeters. Image B shows this corresponding decompressed level, and image C shows the decompressed C6, C7 level. Image A here shows preoperative stenosis at C6, C7 level with an AP diameter of the spinal column of only 6 millimeters. Image B was obtained about 10 months after the surgery. It shows an increase of the AP diameter to 10.3 millimeters with adequate decompression of the spinal cord at the C6, C7 level. Of note, the patient did have a stable mild spondylolisthesis at C7, T1 that remained unaltered post-op. There were no wound complications, late neurological deterioration, kyphosis, or C5 palsy. New postoperative pain occurred in only 1 of our 21 patients, in agreement with Asano and others following C3 to C6 laminoplasty. The addition of dome laminotomies does not alter the favorable pain outcome of C3 to C6 laminoplasty. Socorro and others reported late neurologic deterioration at the caudal segment in 3 of 137 patients, 16 to 60 months after C3 to C6 laminoplasty. None of our patients evaluated at an average of 14 months postoperatively experienced late neurologic deterioration, perhaps owing to C7 dome laminotomies increase in the AP diameter of the spinal canal at the cranial aspect of the C7 lamina. In conclusion, C3 to C6 laminoplasty with C2 and C7 undercuts, i.e. dome laminotomies, was safe, well tolerated, and was associated with satisfactory early and late improved neurological function and decreased pain. Thank you for your attention.
Thank you, Jeff. The laminoplasty is a very powerful technique and is very popular in Asia, but it's probably underutilized here. And one of the reasons is the misconception that it is associated with increased post-op pain. But as Jeff saw, that's not really our experience. We don't see an increase in the pain. On the contrary, we see significant improvement. And we examine whether this has to do with the modified technique that we're using.
Great. All right. Thank you very much, Jeff.
All right.
So then I'll share my screen. What do you see there? We're ready for Dr. Mako. Okay.
Are we looking for Jay?
I think so.
Hello, can you hear me?
There we go. Yeah, sorry, you might have been muted there.
Okay, sorry about that. Well, first of all, what a fantastic and amazing day. To each of the residents and medical students and all of the faculty advisors, it makes me incredibly excited to see all of the high quality research that's going on. Dr. Hambardzumyan, thank you so much for the talk early this morning. It was really inspiring. We have a couple of our earlier residents who are starting to pursue a more basic science type direction with their work. And I think that your talk was certainly an inspiration to them. To wrap up Research Day, amongst all the residents and the faculty and everyone who's been here through this stretch, we have a couple of awards to provide. The first is the Cal Post Neurosurgery Resident Publication Award. Next slide, please. This is an award that was started by Dr. Post. This is one of the honorable mention papers. We have a number of honorable mention studies that there was a lot of discussion about and excitement about who was going to eventually take home the Cal Post Award. But we thought it was worth acknowledging the residents who put out some amazing work as well. So an honorable mention to Dr. Owerman for changing the causes of the United States neurologic disease mortality from 1999 to 2017, which was an excellent paper. Next slide. Another honorable mention to Dr. Yeager and his work in trends in neurosurgery education and training over the last decade. Next slide. And the final honorable mention goes to Dr. Schuber. Respect your elders, the effects of aging on intracranial pressure monitoring use in traumatic brain injury. And with that, we'd like to announce the winner of the Cal Post Award. Next slide. And for dramatic effect, the winner of this year's publication is, next slide, Dr. Rothrock, with Survival Trends After Surgery for Spinal Metastatic Tumors, 20-year cancer experience. Robbie, thank you very much. This is an outstanding study. Normally you walk up to the front, we give you your award and such, but we're trying to figure out how to do this virtually, so that's not going to be the case at this time. Congratulations, Robbie. And then I think we now will move to the presentation winners, where we'll review the Research Day presentation winners. Chris, can you put that up? Chris, you with us?
Sorry, just hold on a second. Some more dramatic effect.
So today, this was an extremely tight race. We had a panel, including our honored speaker and a number of faculty scattered from around the group. While everyone was presenting, we were performing real-time scoring of each presentation. So this is a purely numeric process. Thank you, Dr. Hambardzumyan, for staying on the call and on the Zoom and participating and lending your insight to all of these talks. And as a result, we are going to give away or hand out not handout. We're going to show it to you, but you will get a certificate for a honorable mention, a runner-up, sorry, would be the right term, runner-up to the Research Day Presentation Award, and then a Research Day Presentation Award. So in the first award would be to the runner-up, winner of the Research Day Presentation Award. Chris, can you show that? That's Ray. Ray, thank you very much for your excellent talk. I liked how you adjusted it to the global pandemic, and congratulations on runner-up for the Research Day Presentation Award. And in further excitement and nail-biting entertainment, we have had a tie for the first place winner for the Neurosurgery Research Day certificate and award. And in no particular order, those two winners are, Chris, could you please show? One of our students, Michael Martini, who gave an outstanding presentation utilizing game theory principles and machine learning models in spine surgery. Very impressive talk and very impressive work. And our other co-first place winner is Alex Schuper, who presented on 5-ALA and enhanced surgical visualization of high-grade glioma, a prospective multi-center study. Alex, it was great work. You really started the day off with a bang, being one of our first presenters. We greatly appreciate your work on this project. With that, I'll say a final thank you to everyone who's been a part of this very special day. There was a time not too long ago where we would, struggle to fill a few hours of research presentations, and I'm blown away with the fact that this year, we had twice as many submissions as we actually had presentations. The competition just to be one of the people presenting was incredibly fierce, and it makes me very proud, and I know it makes Dr. Bederson very proud to be part of this department. Thank you all very much, and have a great rest of your day.